and welcome to the sitting of the EFRA Select Committee, looking at the very serious issue, certainly in my part of the world, of seafood mortality off the East Coast. Could I first of all thank the public for their interest in this session? A lot of people have contacted the committee with evidence. The purpose of this session is to hear that evidence, and it will be an opportunity to test the arguments and the possible causes of the mass mortality of crustaceans and the impact on local communities. The committee is not a scientific body, although we do have scientists as members of the committee, so we won't be able to come to a definitive view on the cause, but at the end of the session we will consider what action we think appropriate, which may involve contacting statutory bodies, ministers, etc. So, starting off with panel one, could I ask, starting with Trudy on my left, to introduce yourself and your role, who you represent? Thank you.
Thank you, Chair. I'm Trudy Wakelin. I am the Director of Marine Planning, Licensing and Global Marine with the Marine Management Organisation. We are the body responsible for the regulation and consenting of marine development within English marine waters.
Thank you. Mark?
My name's Mark Rice. I'm Deputy Director for Water and Land Quality with the Environment Agency. Our role is to take out pollution investigations and to work with our colleagues on environmental monitoring.
Rachel?
Thank you. My name is Rachel Hartnell. I'm a Science Director at CFAS, the Centre for Environment, Fisheries and Aquaculture Science, and my role here is to represent some of the science investigations.
Thank you. And Jerry?
Good afternoon, everyone. My name is Jerry Hopkinson. I'm the Chairman of PD Ports. PD Ports is the port operator on the River Tees and the statutory harbour authority for the river and the estuary.
Thank you very much. You're all very welcome indeed. Maybe if I could start the questioning with Rachel from CFAS. Now, the DEFRA-led joint investigation concluded that, and I quote, no single consistent causative factor was identified for the mass deaths of crabs and lobsters, although a harmful algal bloom in autumn 21 was identified as of significant, I think is the word that was used. What evidence points to an algal bloom as a likely cause?
Thank you, Chair. So in the area at the time, there's good evidence to suggest algal blooms, plural. The first bloom, as we understand it, identified both by satellite data and through some testing in the area and around the area, is proposed to be something called Karenia micomoti. This is a bloom that forms very large, dense blooms and was almost certainly present in the area at the time. The data that we have showed that the bloom appeared to persist up until around about a week into the incident, whereupon the temperature of the water dropped and we believe that the bloom disappeared, potentially crashing. When such blooms crash, they can cause oxygen depletion on the seafloor, which may well have been implicated in the incident. There are at least 10 peer-reviewed publications which describe health impacts on crustacea from blooms, and it's recognised that around about 22 species of harmful algae can impact crustacea. We've reviewed the literature, and worldwide, including in the UK and the south of Ireland, there's at least 23 incidents of something similar happening in the past, one in the UK and one in the southwest of Ireland. Now, that alone does not constitute cause, but at the same time, we know in the area that there was another type of algal bloom, the type of algal bloom that you can't see from satellite data, but is the cause of something called diuretic shellfish poisoning, which is a regulated suite of toxins that we regulate for bivalve mollusks from a human health perspective. We tested impacted crabs for those toxins, and we found quite high levels of the toxic compounder, oxidaic acid, in the crab tissue. So we suspect that there was a large Karenia micomoti bloom in the area, which the evidence that we have suggests crashed around about the time of the mortality.
So that one in itself is not a toxic algae?
No, that's correct.
But when it dies and falls to the bottom,
it causes, so as the
deoxygenate.
cells die, so the blooms will die as a result of perhaps a temperature drop in this case. We can't be certain of any of this, but this is the hypothesis. The blooms crash to the bottom of the sea. They take out the oxygen. That causes these localized anoxic conditions. We postulate that, coupled with the diuretic shellfish poisoning toxins, oxidaic acid, that is the most likely cause of this mass mortality event.
And how closely did the area identified through satellite imagery correspond to the area where shellfish were killed?
The resolution of the satellite imagery very close to the shore is not perfect, but it was corresponding with the area.
Okay. Now, some have argued that algal blooms are unlikely to only target crustaceans and deaths of other sea creatures would be expected. So why do you think this algal bloom mainly seemed to affect crabs and lobsters, not fish or seals or seabirds?
So in other instances of this type that have impacted predominantly crustacea, it's theorised that other benthic life that can move away from those oxygen-depleted conditions do that. Fish swim away. The crabs and lobsters have less opportunity to do so. Here, though, we're also saying that the presence of an additional toxin from this second bloom that was in the area at the time, and I think that's what makes it unusual, had an additive impact upon those species.
Understood. We've also heard that larger algal blooms have occurred in the North Sea without causing the associated mass die-offs. So why do you think this particular algal bloom in and after September 21 was so deadly?
Well, and again, we aren't certain about this, and we're certainly trying to test this hypothesis at CFAS at the moment, but it's the anoxic, the oxygen-depleted conditions on the seabed coupled with the presence of the toxin from the diuretic shellfish poisoning bloom, this oxidaic acid, and that's really what we're focusing our work going forward on.
Okay. Could the discoloration of the seawater picked up by the satellites be picking up, for example, dredge material that was washing down the coast that was associated with the dredging of the River Tees?
Yeah, so there's certainly some uncertainty around the satellite data. Our collaboration with the Plymouth Marine Laboratory, who have a model that looks particularly for Karenia micomoti. This Karenia bloom is what we suspect was present here. Our colleagues at the Plymouth Marine Laboratory also corroborated this.
Okay, thank you. Barry Gardner.
Thank you very much. Again to you, Ms. Hart. My understanding is that the bloom in September to October reached levels of 15 micrograms per litre. But in general, is it not the case that a bloom is considered dangerous around 30 micrograms per litre, double that?
So absolutely, yeah. So what we don't have are readings of cells from the area at the time. So we can't be certain of the levels, but there's certainly evidence that is consistent with a bloom. We can't be certain about the density of it.
And can you... sorry, I'm not quite clear what your response to that question was. Is it correct that normally it would be considered dangerous at 30 micrograms per litre?
Yes.
And the evidence such as we have it shows that the blooms on this occasion may have reached up to 15 micrograms per litre.
From my information, what we don't know, we don't have good evidence of the bloom concentration predicted at the time because we don't have cell counts at the time where we predict the bloom.
The submission from the Plymouth Marine Laboratory on that point, you believe, is OTS?
What I would have to do for the committee, if you don't mind, is to go back and check those data. Thank you, and we can submit that at a later date.
Thank you very much. That would be helpful. Now, the image phenomenon seemed to centre on the Tees mouth area rather than the whole affected area. Is that correct?
Yes, although the bloom did spread down the coast to an extent.
But you would confirm that blooms usually occur in bays or inlets rather than in the open sea. Is that correct?
Usually, yes.
And in this case, that was not the case.
Well, the weather conditions, it was very calm, it was very warm. Yes.
Well, when we're talking about the conditions, I understand that although sea temperatures were higher than normal, blooms such as this are actually very rare at that time of year, and the historical record of the temperatures in area are far below the optimal temperatures for a bloom. Is that not also correct?
But at the time of this, although you're right, it is towards the end of the bloom season, we've got good evidence to suggest that there was a bloom of Karenia in the area at the time, which was consistent with the seawater temperatures, which then, as those seawater temperatures dropped quite suddenly, I think by around 2 degrees, did crash, or certainly disappeared from the satellite.
Why was the joint investigation declared closed, given that the findings were not conclusive?
Is that a question for me?
I assume you were part of the closing of the investigation, but I'm very happy if anybody else feels better qualified to address it.
So the investigation was closed because there was no further evidence to suggest that anything else could have been causal. By the time the investigation had closed, we'd been investigating for around about 6 months. It was very difficult then to go back to that environment and test anything else.
The findings at that stage were not conclusive, but equally you had no further evidence to proceed on.
On the balance of probabilities, it was felt that it was the most likely cause. But as I've said, we are testing that hypothesis going forward.
Yes, so in effect the investigation has been reopened.
I wouldn't say reopened, I would say that we were testing a hypothesis.
Okay.
Could Mark maybe add to that in terms of the extent, because you were involved?
The Environment Agency was involved in the initial incident response and doing the water quality sampling and supporting the investigation, yeah. And because we didn't find evidence that water quality caused this issue, then that was handed over in terms of investigation to DEFRA back in December 2021, and the Environment Agency supports that investigation, yeah.
Understood. Sorry, Barry.
No, thank you, Chair. If I could throw a curveball to Mr. Hopkinson. Mr. Hopkinson, on the BBC documentary recently, you said that the subject on the Orca dredger, only virgin sands from the Tees mouth were taken. Is that correct?
Yes, it is.
But that's not true, is it? The sand always contained background levels of contamination, even while the infamous South Bank Wharf was derelict. And there is evidence in the document known as Slab 5, which measured contamination levels of sediment in dredging drop zones around the country. The highest levels were found in Tees Bay, Slab 5, last reported in 2014. So it's not the case that it's always been just virgin sand, is it?
Well, whilst I wouldn't claim that I was seeking some form of licence in my description of the material, what I was trying to make explicit to the viewers of the documentary was that the materials which we had picked up and taken out to the designated spoil ground were materials that by the action of the sea we had previously removed and repeatedly removed on the cycle of climate action, which continuously frustrates our attempts to maintain the navigable channels into and out of the port.
Dredging.
Yeah, we are going to come back to dredging later in a future question, I think.
I'll leave the dredging issue there then for you to dredge up later, Chair. Again, Ms. Hartnell, how likely is it that the further investigations that you're now doing, which are due to conclude in March, I think, of next year, how likely is it that they'll be able to come up with a more definitive conclusion? Is the data that you need to draw further conclusions actually still available?
So the hypothesis that we're testing through our experimental work is that the action of oxidaic acid, the toxin that we know was in the crabs because we measured it, coupled with anoxic conditions at the seafloor, which we can reproduce in our laboratory, are likely to cause the clinical signs that we saw in the animals at the event. Now, we can do that experimentally. It's quite a difficult set of experiments to run, but we can test that hypothesis experimentally. So we will, I'm assured, by the end of March, be able to give a very much greater clarity on whether or not this hypothesis, and I stress it is a hypothesis, in the absence of anything else that anyone could determine, was likely to be implicated here.
If I can just summarise what I think you're... You're saying that you are creating in the laboratory a cocktail of Karenia Mikimotoi, and its collapse, and you're then overlaying that or undergirding that with the other toxin
Not
see if it has the effect that was evinced in the crustaceans.
almost, but not quite. But yes, the principle is the same. So what we're doing is we're recreating anoxic conditions, so the conditions that were modelled. So if we go back to your question about the likelihood of Karenia causing the oxygen depletion via crashing, that has been modelled using previously published work. So with the bloom densities, we have a predicted oxygen depleted level at the seafloor at the time of the incident. The experimental protocol recreates that oxygen depletion condition, so we don't use Karenia to absorb the oxygen from the water, but we do introduce oxidaic acid and the Dionophysis toxins, so these toxins that we know were there, into the animals through exposure and feeding. So yes, apart from we artificially deplete the oxygen in the water rather than using a crashed algal bloom to do so.
And is that because you're convinced that a crashed algal bloom would have no material difference on the anoxic state of the water?
What we're interested in, so in experimental science, you have to really try and control as many variables as possible, I'm sure you know. So we're interested in anoxic conditions, so we're testing anoxic conditions.
If an algal bloom was the chief cause, has human activities such as sewage or pollution and climate change made algal blooms more likely? Can anything be done to reduce the likelihood of further such conditions being prevalent?
Yeah, so, I mean, algal blooms are natural, but they are influenced by human activity. They're influenced by warming water temperatures, so potentially linkages with climate change, but they're also linked to changes in nitrogen phosphate ratios in the water from eutrophication conditions, so runoff, human activity. So yes, but they are also a natural occurring event.
So it is possible in the circumstances that we're looking back at that there were other factors that could have influenced the crash and the subsequent decline of the crustaceans that we saw.
Absolutely, but I think I'll defer to my Environment Agency colleague with respect to potential sewage contamination, which I believe was ruled out as part of the investigation early on.
Forgive me, no discourtesy intended to the panel, but I'm due in another select committee 3 minutes ago and have to leave.
I think Robbie's going to come on to that with his next question.
Thanks very much.
Thank you.
Thank you. Robbie Moore.
Thank you, Chair. I'm wanting to just focus a little bit on the ruled out causes. So maybe Mark, if you want to pick this up to start with, then we'll come to the others. So the joint investigation ruled out potential causes, including sewerage, animal diseases, seismic activity, wind farms, undersea cabling, temperature fluctuations, and severe storms. And the report explains why diseases were ruled out, but says little about the other factors. So putting aside dredging and pyridine, how confident are you to safely rule out other causes?
Well, I mean, the Environment Agency's job is to respond to major incidents like this, and obviously this was a serious concern to the community. So, I mean, we responded immediately on reports of dead and dying crabs. It was a risk-based and hopefully proportionate investigation looking for a cause of pollution. I mean, that's the presumption that you start with as a cause of pollution. You will be able to find someone who's caused that pollution and potentially take enforcement action. The water quality work indicated that that we weren't able to establish a cause of pollution. We looked at lots of different lines of inquiry. We looked at previous reports of pollution incidents in the area. We checked water treatment works and associated infrastructure. We have regulated industry teams. They looked at permit compliance on Teesside with the emphasis on those sites which discharge into the Tees estuary. We've talked about agriculture. We checked around the area to look for agriculture and other diffuse pollution sources. Visual observations on the coastline to see if there was anything unusual. Odour is a good way of picking up potential chemical pollution. And we also undertook a desktop study to identify any industrial sources of pyridine. This was done once we'd identified pyridine in the crab tissue. But none of that indicated the levels of pollution that would cause the type of incident that occurred. So those were the...
Just to interrupt briefly. You're saying that, are you talking about a level that would precipitate an algal bloom through eutrophication, or one that would actually do a pollution incident where the pollutant itself would have been toxic and caused animals to die?
That's right, yes. One that would cause that kind of significant impact on the environment, yes.
Understood. Sorry, I'll be back to you.
So, you were going through potential sources there and ways in which you'd carried out your investigation, but did you consider potential industrial sources?
Yes, we certainly did, yes. As I said, we've got a regulated industry team based up in the northeast area that regularly monitor and ensure compliance with our permits, and so we specifically looked into permit compliance by industry on Teesside with a focus on those sites that discharge into the estuary, and we found no evidence of pollution to back up any of the issues that occurred.
And from your position, what measures are in place at the moment to ensure that construction works do not pollute watercourses or indeed the sea? And are you confident that those are sufficient enough?
Might be something that Judy would be able to answer.
So in respect of the construction which is happening obviously terrestrially, the MMO does not have any involvement in that activity. Dredging works per se, and I'm conscious that the committee may wish to come back with more specific questions, but dredging is an activity that statutory harbour authorities have the ability, the statutory power to be able to undertake under their own legislation. However, there are circumstances in which licensing conditions are placed on licenses in order to protect water column from impacts of surplus materials, and similarly to then consider how the dredge material is disposed of. During the work that was undertaken during the time of this particular incident, it was maintenance dredging which was being carried out under the port zone permissions. The licence which was regulating that work was for the disposal out to the permitted site out offshore.
No, the EA reassured enough that those measures are sufficient.
I mean, the EA is consulted on these issues, but our primary role is in terms of incident response and regulating on onshore sources of pollution as opposed to offshore. Yeah.
Okay, Rachel, do you mind just going back? If I go back to the original question, just which was getting to, which was that the report explains diseases were ruled out, but says that there were says very little about other factors. So putting aside dredging and pyridine, how confident are you that we can safely rule out other causes?
So my organisation carried out testing for aquatic animal diseases, and we are very confident that animal diseases can be ruled out.
Okay. So, Mark and Rachel, I don't know which one wants to come to us first, but so have you investigated whether the deaths of other animals in the area could have been linked to these pollutants?
There's a big thing about dogs dying and people getting ill at the time, you may remember.
Yes, so I do remember that. So previously, dog deaths have been linked to algal toxins. There was no evidence to suggest that in this case, any of the dogs or other animals dying at the time were in any way associated with this event. And that was backed up by veterinary evidence at the time where there were sick dogs who'd not been in any way associated with the shoreline.
And I know the report it obviously is inconclusive, but do you think that there are several factors that could have been that could have led to what's happened rather than just a single cause?
Well, I mean, the environment is a hugely complex thing. We're talking about algal blooms here in the context of being the most likely cause, but even there we're not talking about perhaps just one. We're talking about algal blooms plural, the potential for anoxic conditions on the seafloor and the cumulative effect of another toxin of which we do not know the impact upon crustacean physiology.
Mark, on the same question.
Could you repeat the question?
Just whether just around multiple causes or a single source. What's your view on that?
Well, as I said, our job is to provide the data and the monitoring in terms of water quality, etc. So I don't think that I'm qualified to answer that.
Okay. Jerry, anything that you wanted to add to those questions or any other points?
No, I'm happy to talk about the dredging and the mechanics of what we're undertaking in due course. Thank you.
Fine, thank you.
Dr. Neil Hudson, who is a scientist.
Thank you, Chair. Can I just go to Dr. Hartnell first in terms of the sort of potential multifactorial nature? And it's very hard in science to prove definitively causation and whether there is an association with something or whether something is causing something. So you mentioned that in the tissue samples of some of the crustaceans there were high levels of some of these algal biotoxins. Did you look at control animals, taken, from fishing vessels outside this region to sort of see what the normal baseline was for some of these toxins and some of these other elements and chemicals that you were testing?
No, so we were looking for algal toxins, specifically those indicated by the clinical signs of the animals, so neurotoxins in the first instance. In the UK, we have no baseline monitoring programme for algal toxins in crustacea, so we do not have baseline information.
In that sense, it's quite hard to compare, isn't it? To control animals that are not showing clinical signs compared to the animals that are showing clinical signs.
That's absolutely right. So we're not making that claim. What we're observing in the tests that we did on affected animals and some animals that weren't affected is there does seem to be higher levels in affected animals, but we're yet to demonstrate a statistical significance of that.
And then in answer to my colleague, Mr. Gardner, you were saying that you're going to be testing some of these hypotheses moving forward in the laboratory. And what about, in follow-up to my colleague, Mr. Moore's question about multifactorial nature, have you looked in the literature in terms of other variables that you can actually then say in a multifactorial analysis whether they are involved or not, or whether it's just one particular factor that you've said we're looking at this one and there seems to be a strong association, so therefore our studies will close?
In the first instance, we're looking at anoxic conditions, anorchideic acid, because there is a reasonably compelling theory which suggests that could be causal. When we get to the end of those studies, we, of course, will publish those data, take stock and see what else we need to do.
Right. And is your department, are people working with the university sectors, academics working in this field as well?
So we've had a series of meetings now with our academic colleagues, and absolutely everybody wants to get to the bottom of this. We stand ready and are collaborating with our academic partners.
Thank you, that's encouraging to hear. Right, Chair, I'll carry on with my line of questioning then. So I'll come to Mr. Rice now. Some analysis based on freedom of information requests from the Environment Agency suggests that some crab tissue samples from affected areas contained much higher concentrations of the harmful chemical pyridine than baseline samples taken elsewhere. And I've asked about sort of baseline in terms of some of the other studies. Mr. Rice, Mark, do you accept that analysis?
Yeah, I mean, we certainly carried out analysis of crab tissue. It was, the test that we carried out wasn't accredited for testing crab flesh for chemicals. It was adapted from a water quality test. And so essentially that test can't be relied on to give figures on the levels of pyridine found, just an absence or presence of the chemical. And so if you did look at the levels from the test, the levels of pyridine found in crabs from affected and non-affected areas was fairly well spread. So picking the highest and lowest, it tends to misrepresent the data. So yes, it's true that we certainly did identify pyridine levels in affected crabs, but the technique that we use is not really designed to assess the level in the tissue.
Right, so you're not saying, you detected levels both in the affected area but also in crustaceans sampled from areas outside the affected area, and
yes,
both, I guess the experimental side and the control side had levels of pyridine, but you're not being able to say statistically significant levels of higher levels in the affected area, is that right?
That's right. I mean, we found a healthy crab from the Norfolk Wash had nearly 10 times the concentration measured in an impacted crab from Runswick. So, yeah, there's quite a spread in the environment of pyridine levels in crabs.
But yes. I guess you're talking one crab there. So in terms of numbers, you don't have huge numbers in terms of statistical analysis in either the affected area or in areas where you're not seeing crustaceans showing these clinical signs or death.
That's correct, yeah.
So I guess more work needs to be done then. Would you conclude that?
So yes, if I could pick it up there, please. So I'm conscious from the very beginning that when we're making really important decisions on the basis of test results, the methods that you apply really should be very well validated and, if possible, accredited. And that was the case for methods in water and not, as Mark describes, for the methods that were applied to the crab tissue for pyridine testing. One of the most important elements of the CFAS work commissioned by DEFRA post-investigation is to develop such a method for pyridine in crab tissue. Once we understand the performance characteristics of that method, we'll be able to understand the variability and we'll be able to apply those tests to animals that were impacted at the time, healthy animals now, and really explore what this finding, which suggests that there are levels of pyridine in crabs post-mortem, really means. And that's, if I may just continue for a moment, that hypothesis is part of that which we're testing at CFAS, where the presence of pyridine in the tissues of these animals after death is associated with endogenous production of pyridine as part of the breakdown process, the breakdown, the disruption of cellular processes after death.
Thank you. And I'm just interested, there's a slight difference then between the EA and CFAS. So in the EA studies of pyridine, you're looking at pyridine levels in the affected area, but also you're sampling crustaceans in different areas. But with the algal biotoxins, what you've said to us is that you've sampled it in the area, but you don't have baselines outside. So is that something that CFAS will start to look at in the future to get a baseline in other areas?
Certainly baseline information in any sort of environmental testing is really important.
Okay, thank you. So why was pyridine ruled out by the joint investigation as a potential cause of the mass mortalities then? Is that a fair comment? Has it been ruled out, or are you still open to testing that hypothesis?
Well, in terms of Environment Agency water quality monitoring, both at the time and since, we did not detect pyridine in the water. We have not detected pyridine in the River Tee since 2018, and we can detect it at extremely low levels now, down to 0.01 micrograms per litre. And so the highest levels we've detected in the Tees was back in 2012, and that was at 2.4 milligrams a litre. So I suppose we did not find evidence of pyridine in our water monitoring.
The low levels made you rule it out then?
That's right, yes.
Okay. Would you agree with that, Dr.
I would.
Hartnell?
I mean, there didn't appear to be any credible source of pyridine in the environment. The other thing that perhaps I could add is that the chemical properties of pyridine don't predict that it absorbs well to sediments or that it bioaccumulates in animals.
Further to that, I mean, can pyridine accumulate in sediments and damage sea life in the sea floor?
There's not evidence
Right.
of that to the extent that pyridine is excluded from the European Chemical Agency compounds required for bioaccumulation tests. So it's not considered at the moment. I mean, and we're always mindful that new evidence can come to the fore, but at the moment it's not considered to be a chemical of concern.
That tees me up for my sort of final question of this
okay.
section then. Do you think then that further research is needed to better understand the impact of pyridine on the wider ecosystem?
Well, I think, I mean, the short answer would be yes, but I think the perhaps one of the most interesting elements here is to understand whether or not the pyridine was produced from within the animal itself as a product of cellular breakdown processes after death. Now, that is very interesting. And if I could just elaborate on that slightly, pyridine is a molecule that's in many biological processes. If we think back to our O-level biology when we learnt about cellular respiration, pyridine nucleotides are those which they're the coenzymes which help to produce energy at the cellular level. Now, it is possible and predicted perhaps that pyridine itself is generated to the point where we can detect pyridine and its derivatives after death. And that hypothesis is supported by the use of pyridine nucleotides as markers of post-mortem.
And finally then, are you monitoring the presence of pyridine and also some of these algal biotoxin in animals that prey on crabs and lobsters, but also other animals potentially in the marine ecosystem, picking up seals and marine mammals on strandings or in bycatch? Are you monitoring both pyridine and algal biotoxins?
Right, so if I can take one at a time.
Yeah.
So firstly, there's no monitoring programme for algal biotoxins outside the official control testing for bivalve shellfish, which is a piece of UK retained legislation. When we hear of incidences which are potentially connected with accumulation through the food chain of biotoxins, then we do test as part of our own investigative work. But there's no
any animals, any seals, or any dead porpoises, or anything in the area. No.
Not as part of this investigation, but we have done in the past. But as I say, there's no routine monitoring outside that of bivalve shellfish. For pyridine, we don't in CFAS monitor for pyridine, but the development of the validated test method pointed at that crab matrix, which is quite complex compared to water, will enable us to look further and more for that in the future.
Okay, and Mark, then is there any need then would you suggest that there could be some routine monitoring of pyridine in bycatch or in dead animals, etc.?
In terms of the Environment Agency's responsibility for water quality and land-based sources of pyridine, yes, we do monitor for the presence in the environment, and as I said, we haven't detected it in recent years in the Tees estuary. We also, as part of the investigation, looked at potential sources of pyridine from land-based sources and where it was previously discharged, and neither of those discharges are currently producing pyridine and releasing it into the environment. So in terms of a regulatory role for the Environment Agency, we did that work as part of our investigation.
Thank you. Chair, thank you.
Thank you. Just before we move on to Rosie Duffield, you said you'd picked up very low levels of pyridine in 2012. Could I ask,
yes,
how regularly do you test the water in the Tees?
We have a monthly monitoring programme of
Monthly.
water quality in the Tees, yes. Yeah.
Okay. And just to Rachel again, if you're comparing pyridine levels in a crab that had been dead for some time with pyridine levels in a crab that had been landed as a healthy crab to go into the food chain, could those two tests be sort of seen as comparable, or things that happen after a crab dies could change the amount of chemicals in that particular crab?
Well, yes, again, but the important thing here is to have a method that we can be certain about in terms of its reliability. And that's really what the focus of part of the work in CFAS is at the moment, to produce a method that is at the same level and standard as those that we would use in regulatory testing, so that we can have confidence in the quantities that method returns. And once we have that, then we will be able to examine freshly dead animals, long dead animals, environmentally contaminated animals or not. So it's really about having a method where you can be as certain as possible, certainly when there's so much at stake as there is here, that we've got reliability in those test results, because there's really important decisions being made on the basis of those results, and they need to be as robust as possible.
Thank you, that's helpful. Rosie.
Thank you, Chair. This is more of a broad question to all the panel. Just for a minute, leaving aside the pyridine specifically, could the dredging of polluted sediment just in general lead to the death at this scale and geographical extent, as some university computer modelling has suggested?
I take that one first.
Yeah, good, thanks.
So part of the process that the MMO goes through when we receive any marine licence application for dredging activity is to look at, firstly, a sample plan is required by the applicant in order to determine and agree the number and location and the determinants of samples that need to be taken in order to understand the nature of the material that is proposed for disposal. We use CFAS as our scientific advisors for that process. They refer to the OSPAR Convention, the list of chemicals for priority action, in order to identify likely chemicals of concern, but also use their knowledge of local historical industrial activity, for example, in order to ensure that that sampling suite is as robust as possible. What we will then follow up with, having had those samples taken and analysed, is to consult again with CFAS on the likely impacts of the volume of material, so things like the smothering potential, sediment plumes that could be created, the location of where the material is intended to be disposed of. And there are a number of licensed disposal sites around the coast. Again, CFAS hold a significant database with information from previous dredging campaigns and the likely impacts that could be felt from that. So all of that is considered on a site-specific and on an application-specific basis in order to ensure that we can be assured that the material is appropriate for disposal at sea, but also any potential risks or impacts from that proposed activity can be considered and, if necessary, mitigated through licence conditions, for example, by controlling or restricting the volume of material that's placed, the location or the time of year when it's placed, and how that's then spread within the disposal site.
Thank you. So would that have included, presumably taking into account the decommissioned coke ovens that produced lots of pyridine, just going back to pyridine again for a sec? Would that have been a consideration?
So, as has already been said, pyridine is not on the list of chemicals of concern. So what we are doing as the UK is a signatory to the OSPAR Convention, we have to act within the regulatory framework in order to be consistent with how other European countries undertake the regulation of this activity. So that will have been considered and the samples allocated accordingly. Rachel may wish to follow up with further detail on that.
Thank you.
To be honest, I can't give very much more detail than Trudy's already provided you with. The licensing conditions were met. Using the weight of evidence approach, the conditions were met.
Does anyone else want to come in on that one? Mark?
I suppose the key point to make is that dilution, mixing and dispersion from the North Sea and tidal action on any pollutant and any chemical would indicate that it would have had to have been significant release to have had this kind of impact. If it had have been such, we would have identified that in our monitoring, both at the time and subsequent to that. And as we haven't, we are fairly confident that it wasn't a pollution source that caused this event.
Okay, thank you. I just wonder if this particular event, having been kind of not expected and kind of coming out of nowhere, as it appears to us, do you think that that will change the way that that is looked at in the future? Will there be other things taken into consideration from now on?
So as part of our role, in order to ensure that we are enabling sustainable development, the MMO will take a best available evidence approach. So we are extremely keen and working very closely with our regulatory colleagues and with DEFRA through this process in order to ensure that any additional evidence that comes out of this study will help inform future considerations. But at the moment, from the weight of evidence approach that's been identified to date, there is no further reason at this time to add additional controls onto the current dredging.
Thank you. This one is Mark and Trudy specifically, I think. There seems to be conflicting reports about how polluted the Tees seabed is specifically. Do we know much about the material at the bottom of the Tees estuary and whether the pollution levels are high enough to cause crustaceans to die off on their own? We've kind of covered that, but is there anything else you might want to add to that? I mean, is there anything else that perhaps hasn't come into consideration before?
So there are contaminants within the River Tees. What we know from the long-term data set, and I think we need to remember that maintenance dredging activity has been taking place in the Tees for decades. Yeah. This is not a new activity. What we can see from the trend on that long-term data set is that overall the position is improving. So through natural accretion of cleaner sediments that comes in and overlays historical deposits, that the levels that we are now finding are reducing over time. What we have also found through more recent sampling for activity which is planned to be undertaken, there are other chemicals of concern, and that dredging has been restricted such that the material has to be disposed of to licensed disposal sites on land because it is not suitable for disposal at sea. So there is that ability in order to take preventative action.
Thank you. Mark, do you want to come in on that?
I suppose all I'd add is that, we'll continue to be an active regulator in the Tees area and ensure the various highest standards of environmental protection, and we'll assess all future reports and respond to those as best we can in terms of identifying sources of pollution and taking enforcement action if we find anything.
Thank you. The next question is really for Rachel. Modelling by CFAS suggests that any plumes of polluted material at the dredge disposal site would be relatively confined and could not be large enough to cause widespread death. Was any modelling done for the distribution of sediment at the dredging zone as well as at the disposal site?
So there were plume sediment modelling run using Telmac 2D hydrodynamic model for shallow water. There were 2 campaigns that were examined that were focusing on campaigns in late September using data from the MMO. These were models. They were based on, I think, the assumption that the material was around about 75% silt, and they used data from tidal ellipses and from the National Oceanographic Centre. and the model was run for 72 hours. The results showed that the sediment plume would have moved down towards Redcar in a southeasterly or southern direction, and for the duration of the model it would have remained in suspension. So yes, we saw the potential for that sediment plume to move down across the affected area.
Okay, thanks. Jerry, specifically at the moment, local media has reported that there was some particularly intense dredging before the mass mortality event. Have you looked at whether the timings of dredging match up with the die-off of crustaceans? But also local media have said that campaigners and fishing groups absolutely are convinced that the pyridine and the dredging were linked, and that is what they believe is essentially making their industry pretty, some fishers are saying they're going to be out of business. Have you looked at those reports in the local media or addressed them? And if you don't believe that's true, how can you reassure the fishers that we're going to be seeing in a couple of panels' time?
Yeah, I think it's a very fair question. And we were, when this die-off was initially reported, as a major business on Teesside, we're the largest employer in Redcar. We're hopefully a major driver of providential change in a region which is facing pretty profound socio-economic challenge. So, we view this not simply as an SHA, Statutory Harbour Authority, not simply as a business, but also as a pretty significant player in the local community. In the immediate aftermath of the die-off, we awaited scientific evidence as to precisely what had caused it. And the reason that we did so was because we knew that in terms of the dredging that we had been undertaking, we had throughout that period and prior to it, and in the aftermath of that die-off, been doing what we call maintenance dredging. So if I can just explain, we have licence to dredge in specific areas along the River Tees to specific depths. So long as we don't stray outside of those areas or go below those depths, we're licensed to remove that material. The river accretes rapidly, and consequently we're dredging likely 6 days a week for quite a protracted period, probably 8 to 10 hours a day, 6 days a week, utilising 2 dredges.
And that material's coming from the sea, or has it come in from upriver?
Both, Chair. So the material which is upriver is washed down ultimately from the Pennines, and that largely comprises silt. The material which is downriver is washed in from the sea and largely comprises sand. And we are essentially, certainly in the downstream area of the river, picking up material which we have previously picked up, I suspect, many, many, many times, and redepositing it again back to the dredge disposal site. Sorry, I've now come to your question. So we were clear that we were doing nothing more than our normal maintenance dredging. I went and saw the fishermen in the first instance at Hartlepool earlier this year, late spring of this year. And it was very clear to me that that community had suffered a very, very significant issue. I think you realise any television programme is highly edited, but I expressed my real concern to the guys at that time. The chap said to me, "You've brought in a super dredger. You've brought in this very, very big super dredger, and you've taken out roughly 40-50% of your allowed dredge volumes in the space of 10 days. We watched it. We watched this dredger at work. The dredger that we brought in was exactly the same size as the 2 dredgers in our own fleet. In terms of its mechanical operation, it's exactly the same. It is a trailer suction dredger. It is in effect a vacuum cleaner. It has a pipe that goes down to a drag head, which empties into a hopper and is pumped into that hopper as the dredger pursues a path.
Is that the same as each of the other 2, or the same as both combined?
No, the same as each of the other 2.
Sorry.
So for the avoidance of doubt, a similar LOA, length overall of about 80 metres, similar beam, similar mechanical operation. The reason that we brought it in was because we had a slippage in that area of the river, and we had outage of one of the dredgers under Lloyd's inspection, and I was asked by our harbour office to sanction the disposition of this dredger, and we did so. So we dispelled the myth, and it's understandable. People are upset. They're looking for a cause. They're looking for something to blame. It's absolutely understandable. We dispelled the myth objectively that this was not some extraordinary dredger that we brought in. And then secondly, we looked at the volumes that we'd removed. So we're licensed to remove the thick end of 3 million tonnes, 2.9 odd million tonnes of dredge spoil per annum we can take out. With this dredger, we took out just short of 150,000 tonnes. We did it over a 10-day period. We did it in effect. If we'd been operating our own dredgers, it would have probably taken 20 to 30 days to remove that, as we'd have worked much shorter days and not a continuous period. What we also made clear to folk was that in the area in which this dredger was working, which was approximately 2 kilometres out into the River Tees Bay, so it started its operation within the arms of the River Tees, the 2 gears that protect the river itself, and then went out in a straight line along the channel that ships come in. We are consented, so we're permitted at statute, to dredge to a depth of 15.4 metres. We dredged along that area to a depth of 14.6 metres. How do we know that? Because A, we monitor both the route and therefore the boundaries of this dredger's operation throughout its activity through our harbour office and its VTS, its vessel tracking and control systems in there. And secondly, we surveyed the area as soon as we can get out there after the dredger had departed. It was, I think the survey was undertaken about 2 weeks after the dredger had departed, and the reason that the survey was delayed was because of the wave conditions that we were encountering out there, and simply getting the survey craft into position to take the appropriate soundings. So I'm sorry that's a long-winded answer to a question, but I just want to make explicit to you as to how concerned we are about this, and why I personally, as the chair of the business and as the leader of the business, do not believe that dredging of certainly maintenance dredging on the River Tees, which is what we have been undertaking, is causal to this.
Thank you for your thorough answer. Rachel, I think you wanted to come in, didn't you?
Well, I was just going to add something about some of the pyridine modelling that was also carried out at the time by CFAS. I suppose this is helpful, perhaps, because it's helpful in terms of us really not really understanding how there can have been such a source of pyridine under the circumstances. So we've said before, pyridine has a set of properties which mean that it's buoyant, it floats, it's highly volatile, and it's soluble. So what that means is that it isn't thought to bioaccumulate in the way that we would see with other chemicals, and it isn't thought to readily absorb to sediments. So that's quite important here. Now, as I say, there may be other evidence that suggests that internationally the chemical agencies have got this wrong, but certainly that is the thinking at the moment. So where we looked at pyridine modelling, so that's to look at how a chemical released into the environment might act in the environment. That was done at CFAS at the time using 3 modelled concentrations of pyridine from 100 to 10,000 litres of the chemical in the disposal site. And the model predicted that over 40% of the compound would evaporate from the surface of the water. The rest of it would be mixed into the water column, and the enormous dilution factors made it very difficult to understand how exposure to pyridine could be singularly causal here. We're not saying that pyridine isn't toxic to marine organisms, but what we are saying is that the concentrations it's very difficult to see how we could have not, with the Environment Agency tests, have picked it up in the water if it was released at the site and dispersed along the coast as it may have happened.
Even at the disposal site, let alone 25 miles away.
Yeah. Thank you. Just a tiny bit more. Yeah. Are all of you confident that you can rebuild relationships or trust with the fishers? I mean, we'll obviously get the chance to ask them directly in the next couple of panels, but are relations at an all-time low, or do you feel that there are conversations and that they're at least confident that you are really looking at this and listening to them and hearing their instincts or their belief in an industry that they are absolute experts in, which obviously you are in your own way, but is there any way of kind of coming together on it? I don't know who wants to answer that.
Can I answer first, maybe? I've now had 2 long sessions with the potters, with representatives from both Hartlepool and Whitby, and I absolutely get the issues that these guys have faced. I mean, one of these guys, I remember Pinky saying to me, "When was the last time you got paid?" Because I didn't pay last month and I didn't get paid the month before. And there's no satisfaction in listening to a man say that to you. You can't help but question your moral compass if you're going to go and sit in front of people and seek to mislead them. You're going to hear from one of the guys, hopefully, a little later. He's a guy called Stan Rennie. Stan and I wouldn't say we've got to know one another, but I would say we've got to know the measure of one another. We've both reflected, as relatively old guys within the industry, that he cannot recollect anything like this happening in the 48 years that he's been potting off Hartlepool. And whilst I've only been on Teesside for the last 15 or thereabouts years, I can't remember a similar incident to this in my tenure on Teesside or indeed on my time in the port industry, this type of incident. Do I believe that maintenance dredging is the cause of this? No, I do not. Do I believe that the guys encountered an issue? Yes, I do. Have we definitively identified the cause? No. What we've identified is probabilities. Do we need to be extremely cognizant of this going forward? Yes, we certainly do. In respect of maintenance dredging, from my perspective, in terms of the governance of our business, it is an absolute imperative that we totally stick to the remit, both in terms of scope, extent and activity that we're given under statute. It's a precious privilege and frankly we should not abuse it. Do I believe that if we undertake capital dredging on the river, and we have plans so to do, whether that's we've got a great scheme to bring liquefied natural gas onto Teesside. We've got a fantastic scheme to develop a deep water container terminal in our port infrastructure. Do we need to look very, very carefully at the permits and consents? Yes, of course we do. And will we join that process? Yes, of course we will. There is absolutely no ideological or logical purpose to operating a business, and we're a successful business, that harms our environment in its operation. So that would be my position on this for PD Ports.
Thanks, Rosie. To Geraint, I think we've covered one or two of the points that we've discussed. You raised.
Well, I've got a few points to make myself, but before turning to Jerry on the dredging, I'd just like to clear a few things up with Rachel on the science. I mean, first of all, can you just confirm that pyridine kills crabs?
Pyridine in certain concentrations is toxic to crustacea.
Yeah, exactly. And you'll be aware of the emerging evidence from Newcastle University that in very small quantities, pyridine kills crustaceans, in particular crabs and lobsters. That's correct, isn't it?
Well,
so we're just clear on this.
I have heard through conversations with our academic collaborators in Newcastle and elsewhere that that's what their
right, that's
results,
all you've. Oh, right, that's all you've troubled to find out before this inquiry, when it's been all over the media, the research they've done and the testimony, and you just said you've heard something.
If I may, excuse me, we have had presentations from our academic colleagues on the data that they have produced so far.
Right, and it kills crabs.
And that certainly is what's being reported. What we haven't yet seen, and we are very keen to do so, is to see the full reports of that work. Certainly I think that it'll be very important to have thorough peer review of that report, and we're really very keen to work with the academics on the work that they're doing.
That's not
okay,
being
so
published
if
yet.
I can go on, Chair.
Yeah.
Secondly, other than that pyridine kills crabs, you'd confirm that pyridine dissolves very quickly with contact with oxygen and water. In other words, if it's dredged up, it's very quick that pyridine then disperses, and that if you were to test it, as Jerry's mentioning, 2 weeks later, in all probability it would no longer be there. The killer would have gone off in the night. That's correct, isn't it?
It is, and I'm going to defer to the Environment Agency on this, if I may, but the pyridine does dissolve in water, and it is volatile, so yes.
Yes, so it does dissolve in water. Okay. And I will come back to Jerry in a moment, but if I may just pursue the scientific questions with Rachel so we're clear. So we know pyridine's toxic to crabs. We know that it was churned up, it sort of disappears quickly. And we also know from Newcastle University, as I understand it, that they're finding pyridine 20 centimetres below the surface of the sediment
And there's concern, therefore, that as you dig deeper, there'll be greater concentrations from industrial waste, from coke ovens from which it's produced. That's true, isn't it?
But certainly that's what's been reported so far with the work that's happened at Newcastle, yes, and I know we'll be hearing about that more later on.
Right. Okay. Now, turning to Jerry on the dredging side, if it's the case that crabs and lobsters sort of tend to, walk along the bottom of the ocean to a certain extent, because they don't sort of swim around like fish, and if it's the case that pyridine is toxic to crabs and lobsters, and also the case that it's found at a 20 centimetre depth that then disappears, you've just testified that you've been digging on your maintenance at 15.4 metres, which is a much greater depth than 20 centimetres. So would you accept that given that you did your surveys 2 weeks after dredging, you could have churned up an enormous quantity of toxic pyridine, killed enormous numbers of crabs and lobsters, come back 2 weeks later and said, oh, where is it? And the answer is it had dissolved.
First of all, my apologies. You may not have understood what I said. I said that the depth that we are consented to dredge down to in that particular area where the UKD Orca was operating was 15.4 metres. You've heard the 15.4 correctly.
14.6, I think you said you did. You were consented for 15.4, you did 14.6 metres, and I'm saying at 0.2 metres, there's pyridine being identified by widespread sampling from Newcastle University.
Well, the site. I'm not genuinely not ducking the question. The answer is, I don't know. I know that the material that we took out, we are permitted and consented to remove, and coincidentally, and it is coincidental, to the completion of that dredge, we surveyed the area to ensure that the depths that we'd achieved were what I'm telling you, and shortly thereafter, sampling was undertaken by ourselves, which was submitted through to the MMO of the dredge sediment areas in line with our triennial sampling regime.
And in that dredge sediment sampling, how much pyridine was found?
I would have to defer to the scientists.
Who's that? Sorry, I'm not sure who we're talking about. Is it? Because Mark Rice, you don't, this is above your pay grade, isn't it? This level of analysis, you just do water quality. Yeah? Well, I'm not trying to be rude, right? But I'm just what we want to establish is whether there's concentrations of pyridine in the dredge from coke that's been identified. Because as I say, Newcastle University, who are experts in this, have found it at just 20 centimetres, and if there's a lot deeper, then obviously it's going to cause a lot more mass death, isn't it? So is there any further light anyone can shed on this? No. Okay.
Who
now,
did you submit this?
What?
Who did you submit the samples to? To MMO or to Environment Agency?
Well, the process is that the samples are gathered. They're tagged. There is a probity system to ensure the integrity of the samples. They go through to CFAS or a similar governmental agency for analysis. Those results are submitted to the MMO, and we are given the, if you like, the green light to continue to undertake dredging.
In addition to this, my understanding is that the sampling, there is some sampling, maybe you can tell us a bit more about it, from the Tees itself.
Yes.
But there isn't sampling from the licensed dumping site. So there's this stuff dumped en masse and it isn't being sampled there. That's true, isn't it?
So
Trudy.
the material is tested in advance of the dredging being undertaken. We also have an annual monitoring survey where we are reviewing a selection of licensed disposal sites around the country. We tested the Inner Tees disposal site in April 2021, and we also tested the Outer Tees disposal site in October of 2021. The April report is now published and confirms that, and again, we commissioned CFAS to undertake that survey work for us, and there were no issues of concern raised during that. The October 21 report isn't yet published, but from the draft report that we have seen, again, we have found no indications of any concern of contamination levels at the disposal site.
So we're clear here. Are you saying to us then that when the Port Authorities do dredge at 14.6 metres into sediment that Newcastle University have found pyridine at 20 centimetres, that you test that and you haven't found any pyridine? Have we got that right? I'm just so I'm not confused.
For the purposes of the marine licence application, the sample plan sets out the suite of determinants which are required to be tested, and pyridine is not one of those elements.
Okay. So you don't test for pyridine.
So
That's
it wasn't
okay.
tested.
So it hasn't been tested.
It wasn't tested ahead of the incident occurring. The sediment has been tested subsequent to the incident occurring, and pyridine was not found.
Is that because it dissolves in water and it has been churned up, do you think?
I would have to defer to the scientists to relate you to the
All right, so it
methodology.
might have been. Well, Mark, have you got anything to say on that? It does dissolve. It just sort of disappears, doesn't it, after killing crabs?
Well, I mean, as I said, we take routine monthly surveillance of water quality in the Tees. Chemicals, nutrients dissolve oxygen, chlorophyll and phytoplankton, and we've been doing that for years, and we've found no evidence of significant levels of pyridine in the environment, and that's what our water quality monitoring programme demonstrates.
Because the worry here, and the issue here, is obviously the natural environment, namely the sea, can absorb certain amounts of pyridine in a safe way to crabs. And the issue here is that if there's a sudden amount of dredging occurred, and I think, correct me if I'm wrong, Jerry, you said that the impact of that you did, whatever it was, 150,000 tonnes in 10 days, where you'd normally do 30 days, which I've got the approaching.
Yes.
So you did quite a lot all at once. And the issue of the nature of pyridine is that it'll come up, it can kill a lot of crabs, but if you do it more slowly, it'll basically sort of, it'll dissolve, and it won't be problematic. And the concern here is that the speed and depth at which the dredging was occurring was an allowing a concentration of pyridine to occur that would lead to mass deaths in crabs. So can I ask just about the specific method of dredging? In particular, do you use what's called remediation dredging they use in Belgium? I think you described something akin to it, which is about having a sort of vacuum cleaner attempt to dredge up a material that then is not allowed to dissipate. And do you also use metal fencing, the so-called curtain approach, sheet pile walls? So the idea would you'd have walls, and then you'd have a suction, and then you'd minimise the prospect of contamination and leakage of pyridine in particular. Do you use those methods? And if not, why not?
No, we don't use a method of containment around the drag head. Our view of the amount of disturbance that this causes in terms of the action of the drag head across the seabed is minimal. I'm certainly more than happy to look at any containment mechanisms, but having talked to colleagues internally about this, they're satisfied that the methodology that we're undertaking is in accord with good practice.
Okay, and finally, the dredged material that you dredge up, which has got pyridine in it, is that all put on land, or some of that simply dumped in the sea?
No. The material that we're dredging up in terms of maintenance dredging goes to an offshore disposal site, which is the one that has been mentioned by Trudy. Can I just make one other comment, please, in answer to your suggestion that this intense operation, which replaced an activity that probably would have taken 20 to 30 days for us to undertake, was somehow causal to this. whilst I would not suggest to the committee that we are bringing in a dredger to supplant the operation of our own dredgers on a regular basis, this is by no means an irregular activity. To bring in dredge support to undertake dredging where we've had significant slippage in the river channels into the River Tees. Now, I can supply the committee with further information with regard to precisely when we've done this in the past, and I would ask you to reflect on the fact that we have not, back to my earlier assertion, in the space of 48 years, undertaking maintenance dredging in accord with the methodology that I've described, without the added sophistication that you've alluded to, but in accord with what we believe to be best practice. We have not had an issue like this on the River Tees, which is why I continue to assert that the maintenance dredging operation on the River Tees has not been causal to this incident. It is coincidental that the dredger arrived at a time when these crustacean mortalities occurred. But frankly, I think we're looking in the wrong direction.
But so I'm clear there here. You do accept that pyridine is toxic to crustaceans and that there's pyridine in the sediment and that it was quickly churned up and obviously it dissipates. But are you asserting here that there's no relationship between the dredging up of pyridine and crabs and the death of crabs, on the basis that crabs didn't die before, so they can't be dying now?
I'm asserting that in terms of the maintenance dredging that we were undertaking, we have done nothing different to that we've been doing for the last 40 to 50 years on the River Tees without these issues occurring.
Sorry, I'm a bit confused because I thought you said you did a lot of accelerated. You did 30 days work in 10 days, 150,000 tonnes, so that is different, isn't it? And I'm asserting
yes.
that if that, because pyridine dissipates, if that generated the concentration of work, generated an extra concentration of pyridine, that might have tipped it over into a mass death situation. So it was something you were doing differently that could explain the deaths, isn't it? That's true. I know you're saying no, it couldn't, but that's true, isn't it?
No, I'm not going to say no, it couldn't. What I'm going to say to you is that this type of relatively intense dredging was not a one-off occurrence. We will have done this many times over the last 40 or 50 years, and the issues that we have encountered in terms of the mortality of crustaceans have not previously been experienced.
And in light of what's happened, do you think there's a case for a precautionary principle from you, namely that you actually do slower dredging, you have more mitigation, with these, steel shields or whatever, and obviously use remediation dredging? I know you said you'd looked at it before, but in light of what's happened and the emerging science, do you think it would be reasonable from a precautionary point of view to now do things a bit more slowly and take a more, more proactive precautionary approach?
I think we're extremely proactive as an organisation. I don't think the committee, with great respect, should form a view that this is an organisation that doesn't care about the environment, does not care about the impact that our activities have on people, and does not constantly review the operating methodology that we're following. Can I just illustrate that?
I'm sure you do care. I'm just saying. I'm suggesting you should do more in light of what's happened.
Can I give you an illustration of that, perhaps, which is a piece of best practice that we've adopted in the past few years?
Yeah, well, I think we're probably running short time. I'm sure there's lots. I'm just wondering whether you do a bit more best practice in light of what these things I've just mentioned, in light of what's happened.
Sure, we'll continue.
Okay. And finally, because the chair will tell me to shut up in a minute, I'm sure. Can I just finally ask Rachel? Is it that obviously there's been a theory put forward that this is an algae bloom killing crustaceans, and I of course accept that algae bloom could and does cause death of crustaceans. What's also been said is that a sufficient reason for the deaths could be pyridine. Assuming both of those are true, if there was a conjunction of those two impacts at the same time, would that help explain the level of death that we've seen?
I think it's difficult to say. I think one thing that I would draw our attention to here, though, is the huge spatial extent of the impacted area, which I think is interesting in terms of the pyridine hypothesis, because the spatial extent and the duration over which this very regrettable incident appeared to take place. So it was really 2 months plus where we were seeing animals dying. Now, it's quite difficult, given we haven't got evidence of a point source of pyridine that was identifiable in the environment from the EA's testing, and we know the chemical properties of pyridine from the European Chemicals Agency, which say all those things that I won't repeat now. The persistence of this compound in the environment at levels that would be likely to impact the crabs, it seems very difficult to understand how pyridine, if it were in the environment, at levels which could have been toxic to animals, recognising that it's not thought to bioaccumulate, so the crabs would have to be almost actively seeking it out, how it could have persisted for long enough over a wide enough spatial extent to be plausibly linked to the entire incident in this case.
We're not talking about persistence here. We're talking about an explosion of pyridine, right, through dredging, suddenly getting into the ecosystem, into crabs and lobsters, and them dying, right? We're not talking about some sort of, a complexity here. That's possible, isn't it? And in fact, very probable.
I don't have evidence to suggest it's not possible, no.
Okay, so you don't have evidence it's not possible. Okay, I'll leave it there.
And just in terms of concentration, given that the pyridine does volatilize, as we've heard. So, if pyridine were killing crabs 25 miles down the coast, what were the concentration levels likely to be at the dumping point of the dredge material?
High.
Dilution.
And I would have to get back to you with a number, a modelled number, but they would have to be very high.
It's worth pointing out we will be doing additional water quality sampling around the proposed dredge areas between October and December this year, so we will have a better picture of additional monitoring in response to what's happened.
Isn't the problem that there was pyridine that it just sort of leaves the scene of the crime? This is the point about it, isn't it? So if you do your monitoring now, what happened months ago, obviously they won't be there anymore. The issue is, what was the concentration at the time of deep dredging on an enormous scale that tipped the balance in terms of the life expectancy of crabs and lobsters? But I'll leave it there, Chair.
Thank you. Ian Byrne, this is the last session in this
thanks, Chair. I just want to pick on something there Jerry mentioned before about the economic driving factors as well behind the Freeport. And I've been on this committee since 2019, since I got elected. I've never had so many emails from the North East region of concern on this topic. So a lot of the emails, the flavour is that the writer feels that the economic and political capital invested in the Freeport agenda by certain leaders means the environmental impact is now a secondary consideration to the rolling out of this project. Is that a fair analysis?
I've read very similar articles, Ian, and I've reflected on this quite a lot. To answer your question, and I'm answering your question as a layperson. I'm not a politician. I'm an apolitical guy running a business. I know how important the levelling up agenda has been to people living in the North East, irrespective of which side of the political divide you sit on. I know the levels of expectation on Teesside have been enormous. I know that the Freeport has been projected as a panacea in many ways, and you may know that I've gone on both national and local media to say that the evolution of a Freeport on Teesside is definitely no silver bullet for the region. I feel that for some kind of collusion to have taken place to somehow suppress this issue on the basis that it could be damaging to a government agenda. With great respect to my professional colleagues sitting here, does huge injustice to, I think, a lot of very earnest scientists and environmental specialists.
There's been a shutting down of evidence, and
yeah, I do.
You know, when we've got the next panel coming along, and when they're saying in their learned manner what their research is, it's getting shut down, and people are looking at that as purely political. So that's not a good place to be, because if there is evidence that needs to be accepted, regardless of where you are with the Freeport agenda or not, we've also got to protect the environment as well. It can't be all So, you know, I'm not casting aspersions on the panel. I'm just saying, through my emails, there's lots and lots of people who believe that it's this agenda and nothing else matters.
Yeah, but you know, Ian, I kind of common sense leads me to question, bearing in mind the whistleblowing policies that exist across the civil service and across a business like mine. With, you know, in my business, direct recourse to my owner in Canada. You know, if people feel that they are being coerced or doing something wrong, they do. I think so.
Okay, thanks very much for that answer.
Yeah, thank you.
Now, I'm going to go to Trudy now. How can we be sure that future capital dredging works, particularly for the new Freeport on the south bank at Teesside, are safe? And, Trudy, can you just, for people watching, they've probably been blinded by science the last hour, what's the difference between maintenance and capital dredging?
Okay, thank you. So firstly, I think it's important to remember that the dredging that was undertaken at the time of this incident was maintenance dredging, which was not connected to the Freeport development.
Yes.
So the Freeport development was only consented earlier, subsequent to that incident. I think it's also important to remember that the Marine Management Organisation is a non-departmental body specifically set up in order that there is an independence around the regulation for marine development that takes place. And also, just to go back to Ms Duffield's question earlier on relating to relationships, the Marine Management Organisation also has a role as England's fisheries manager. So we work day in, day out in managing the activities of fishermen, and it is of absolute imperative urgency to us that we understand what has taken place here, indeed as it is for the whole of the DEFRA group, which is why the investigation was set up and why everybody has worked so hard and so collaboratively in order to try and understand what's happening here, because we all have a role in protecting the marine environment. In terms of the difference between maintenance and capital dredging, to put it simply, because I'm not a scientist, I'm an engineer by background, there is, for the approaches to a port through the navigable channel, or indeed for the berths within a port, there is an accepted depth to which that needs to be maintained for the safe navigation. So underkeel clearance to ensure that at all states of the tide, vessels that are using that port can access. as the term maintenance implies, so that level is set, but because of the natural accretion which takes place, both from upriver sources as well as from the sea, those berths, which tend to be slightly deeper than the surrounding area, the berth pockets, need to be regularly maintenance dredged in order to ensure that that depth is maintained.
Jerry's outlined that, yeah.
So that tends to be, as I say, the cleaner deposit material that's coming in, and the underlying bed is not impacted or affected. For capital dredging, and there are kind of two definitions that we use. One is that any dredging which has not been undertaken in excess of the last 10 years, so it can't be claimed to be routine, or that is increasing the overall depth of the approach channel or of the berth pocket. The regulation which is in place to look at capital dredging, therefore, is more thorough. So we would look at very carefully, again, what the likely determinants might be required to be sampled, given the historic nature of potential contamination from different industries which might taken place. And that would influence the sample plan that the applicant would need to submit and comply with in order for us to be reassured that that material would not be harmful and that it could be disposed of to licensed disposal facilities at sea. As I've mentioned earlier, so the OSPAR Convention is the oversight body that looks at material disposed of to sea for the whole of the North Atlantic and the signatories to it. And the UK is a signatory to that. So we have to be compliant and consistent with regulation which is imposed accordingly. We also have to provide returns to OSMAR on a 6-monthly basis, which sets out what has been done, where and to what volumes, in order that that data can be collected. As part of the sampling process, if any material or any of the samples which are taken exceed what are termed action levels, that prompts further consideration. And there are 2 action levels that are used, not for the full suite of determinants, but the first one being action level 1. This is of concern. We need to consider whether or not it's appropriate in the local environment. That's where some of the site-specific consideration comes in. And action level 2 would be, this is too high, it cannot be permitted to go to sea. That doesn't mean that it can't be dredged, but in those circumstances, refusal to dispose of the material to sea results in then the need for the material to be taken and disposed of to a licensed disposal facility on shore, which is then moves into the terrestrial regulatory regime. That's what is happening at the moment with material which is being dredged at
That
South.
sounds really, really comprehensive and extremely professional. But with the uncertainty that we've heard in the last hour and a half about what the causes of this potentially could be, are you altering your scope of materials that you're testing for? You touched on before, you hadn't tested for pyridine, now you are. You know, is what's happening here making you review actually what you are reviewing, what you're testing for? Because I think I'm really worried about, obviously, there's no definitive, nobody knows here what's happened. You know, we'll have another panel saying a different thing in a minute. And it'll be debunked by Rachel to a degree. So my worry is, can we definitively say, with all the uncertainty that we're actually talking about, is capital dredging safe in that area?
With the best available evidence that we currently have, then my answer is yes, it is safe in that area. But as we've said, we continue to follow up on these lines of inquiry that have been exposed through the work that's been done to date. We are very happy to collaborate with the work that is being undertaken by the academics in order to share the data that we all hold together in order that we can determine whether or not there is changes that might be required either to the regulatory framework or to the protection, the action levels that might be required.
Okay. Well, that's a partial answer, I suppose. If I just touch on, you touched on it before, but also lots of concerns about dredged material from the South Bank escaping back into the Tees. So what controls are you putting in place to prevent this? And how would this material be disposed of in the future? And again, the uncertainty around what's within that material, what it could actually potentially do to the marine environment, which you rightly said was your priority to protect. How are we going to ensure that that actually happens?
So there are a number of licensing conditions which are applied to the consent, which has been granted to the applicants. They control, again, as I've said, the volume, the location of disposal, where material can actually be dredged from for disposal at sea and where it cannot. We have our compliance and enforcement local staff who monitor the dredging whilst it is taking place in order to ensure compliance with those conditions. Was that real time?
Because obviously the line of evidence Garab was going at was the instantaneous disappearance. Is it, what are we ensuring that with this, with this uncertainty about how long it actually stays within the water, so we can actually monitor it?
So for the incident which took place, or that occurred last year, we undertook, as part of the DEFRA-led investigation, we reviewed the activity which had been undertaken in order to ensure compliance retrospectively. Part of the process that we have got in place for the current works is that we are working very closely with the contractors so that we know what they're doing, when they're doing it. They are supporting us with the monitoring that we are undertaking, and we are inspecting both the dredging activity and the disposal activity.
Dredging now, is it?
Yes.
Okay. All right. So if I just, because obviously this year, because it's been a long session, I think, and I'll give this to Jerry first. So with regards to the uncertainty, we know that there's councils that have actually, called for a halt to the capital dredging until we get more evidence and so we get a clear understanding of what's actually happened. Should a ban on dredging be put in place until we've got absolute certainty of what's happened? I mean, capital dredging. What's your thoughts on that, Jerry?
As Trudy has said, Ian, the process is that these guys are being statutorily forced to follow in respect of the grab of the material within an enclosed bucket, the movement of the material onto land and the disposal on land. And I think equally, Trudy, sticking within the curtilage of the dredge area, as long as they are following those, and they're following those rigorously, and I've absolutely no reason to suggest that they aren't. I think it's questionable. I think that what we need to have is a very definitive view as to why it should be stopped. The crustacean die-off did not occur when these guys were undertaking dredging on South Bank. There was no activity in terms of that dredging taking place at that time.
When did that start?
Crikey, I guess a couple of months ago.
Well, September?
No, yeah, it'd be about then. So
we're still assessing any impact. What you're talking about?
Yeah.
Okay, so just to finish, what do you see as the main weaknesses in the dredging arguments that we should put to the next panel? Is there anything that you want me to bring forward to the next panel? That's probably for Trudy and Jerry to finish. If there's not, it's fine.
No, nothing from me, thank you.
I've already said to the committee, the historic nature of this, the continuity of the activity that we're undertaking. I realise I couldn't give examples of the improvements that we've made, but they are improvements. I assure the committee.
You can always put that in writing, Jerry.
Sorry?
You can always put that in writing to the committee.
Can do, by all means.
Okay.
Thank you very much.
Can I ask one very short question?
Well, we really are very, very up against time because we've overrun by about half an hour already. But if it was a short sort of
Very,
yes,
very
no,
short.
sort
I
of
just
answer.
want to, just as a factual question, I just want to ask Jerry,
Yeah.
as we move towards the free port, and
Yeah.
you've talked about this, the history of the dredging hasn't caused problems. As we move towards the free port, is the dredging activity going to increase significantly and therefore, in my mind, increase the risk?
Yeah, that's a great question. The short answer to that is there will be a peak in activity which is ongoing at the moment, which is the capital dredging that Trudy's already alluded to. Once that capital dredging is undertaken, then we're into maintenance dredging to maintain the navigable channels into and out of the free port. I think there could be a minor increase in terms of volumetric amount withdrawn annually. If you'd asked me to make a guess, I would say circa 100,000 tonnes of material, which is a big number, but it's set within the context of 3 million tonnes. Yeah, I think dependent on how successful this is, if the free port, in particular in terms of the offshore wind sector that South Bank is focused upon, can get some of these big contracts. These vessels are fairly big and often can be quite deep in terms of the draft. So there could be an increase, but it won't be, this won't be a seismic shift in terms of the volumes that we'd anticipate having to dredge.
Okay,
Thank you very much.
Sorry, does that answer your question?
Are there any final
think so,
points
yeah.
any member of the panel might want to make before we finish this session? Rachel?
So if I may, and thank you so much for all of your time this afternoon, I just wanted to add one last thing on pyridine, if I may, which is being now spoken about as the calls here. We talked at the very beginning about the uncertainty of the test results, and Dr. Hudson, you indicated the small sample sizes of the animals that were tested at the Environment Agency initially. I'd just like to say again that even with the uncertainty of the test method, pyridine was found within the range of affected animals in animals from outside of the site. So it's really curious that we were able to find evidence of pyridine in animals that were apparently unaffected and a very long way away from the Tees. So I just wanted to draw the panel's attention to that. Thank you.
Understood. Thank you for putting that on the record. Could I thank the panel? This has taken longer than we anticipated, but I think that's no bad thing because it is important, particularly as it involves my constituents in Whitby, that we, test the evidence and we get as much information as possible. Thank you very much indeed. We'll now move on to the second panel, please.
Thank you.
Thank you very much. Thank you.
No, thank you.
To be honest, this 100,000 tonne increase, I don't know what it means as 5%, 10%. I don't.
Check with Mr. Dearjohn. The vote may not be straight away.
We do check.
Yeah. Well, order, and welcome to our second panel. We have two witnesses here in person. We have one online. I think Mr. Dear Jones, could we just check that you're hearing us and we can hear you? Can you hear us?
Yes, I can hear you. And
Wonderful.
Can you hear me?
We can. Wonderful.
Good,
so could I ask our witnesses to introduce themselves and basically what their role is in this matter, maybe starting with Mr. Dear Jones, who's already spoken briefly.
Hello. Nice to meet you all, committee members. I am a marine pollution consultant. I think I sent a CV, which...
yes, we're broadcasting this. Not everybody watching will have read the CV. So if you just like to just point out briefly...
I've been working as a marine pollution consultant since the 1980s. I trained at Cardiff University, where I did do a modular degree in maritime sciences, which was aimed at producing managers of marine pollution incidents. I'm not a biological chemist or a chemical analyst, but I will admit to being a generalist. I was taught research methods as part of my degree course, and so there I am. I have a long history of working in the marine environment on pollution issues and a number of dredging issues to boot. Joe Redfern of the Whitby Commercial Fishermen's Association contacted me in the autumn of 2021 on the basis of having read a document of mine which was lodged in one of the local libraries, where I had done a review of the available data on Tees pollution back in 2007, at which stage it was being recognised as possibly the most polluted estuary in England.
Okay, well, we'll come on to that during the evidence,
that's me.
I'm sure. Thanks for that introduction. Dr. Gary Caldwell.
Good afternoon, Chair.
Welcome.
Good afternoon, Committee. Thank you for the opportunity to come and speak with you today. So my name is Dr. Gary Caldwell. I am a senior lecturer in applied marine biology at Newcastle University, where I've been working for the last 18 years. And to put my credentials on the table, my specialisms are in marine ecotoxicology, marine poisons and venoms, and naturally occurring toxins. I have got a long history of working in the marine systems and understanding the nature and risk of toxicity that human activities pose to it.
Thank you. I'm also a science alumnus of Newcastle University myself.
Yes,
so
I understand.
Welcome, doubly welcome. And Dr. Rodney Forster.
Thank you also to the committee for inviting me to give evidence. So Rodney Forster, Director of the Hull Marine Laboratory at the University of Hull. So my role in this inter-university cooperation, which also includes the universities of York and Durham, who couldn't be with us. I'm looking at the algal bloom hypothesis and reworking and looking back at the original images. That's one part of the work. And the second part is with a little bit of funding from the Fishmongers Company to start fieldwork this year in 2022 out in the North Sea to try and increase our understanding of the northeast coastal system.
Thanks very much. And if you agree with the evidence that one of your colleagues has given in this session, don't be frightened to say I agree with him and move on. But if we could start, I suppose it's the essay question. So the joint investigation led by DEFRA concluded, as we've heard in the first session, that an algal bloom was the likely cause. Do you accept that an algal bloom may have played a role, and if so, how central a role in this particular situation? I think Dr. Forster might be the best person to start with.
Yes, so yes and no. So some of my work supports those ideas and some of it contradicts it. So well, first of all, I should say that number one cause of shellfish mortality events globally is generally algal blooms, red tides, associated often with low oxygen events, but also with direct toxicity. That's probably the main cause, so there's an indication that we should be looking in this direction. Where I came into the investigation, I think we saw the first draft of the report from CFAS, the Environment Agency, around about March or April this year, and the first thing that I noticed, they had a map of chlorophyll concentrations of September last year. And I looked,
was the Plymouth University satellite image, was it?
Plymouth was mentioned. I think that came in later on. So anyway, I looked at the satellite images. Chlorophyll concentrations were, in my view, too high to what I would expect in the northeast coast. The other thing was that period just before the event, up until October the 6th, it was very cloudy, so the satellite image database is not very good. So bear that in mind. It's not a comprehensive view of what's happened. We need clear skies to see the sea surface.
Yeah. Sometimes difficult with these images, when somebody's drawn a line around it, it's hard to actually tell definitively whether it's as clear as maybe having the line drawn around is.
I've gone back to the same information, which is from the Sentinel-3 satellite. It's a European satellite, very high quality, reliable. I've worked with it for quite a long time, and it's the base of the Plymouth and the CFAS work as well. So through the month of August 2021, we've got a very settled situation. It was quite a warm month, very, very low chlorophyll, no sign of any blooms. And I'm looking down the northeast coast from about Lindisfarne, Holy Island, south to Scarborough, that zone that I'm calling the northeast coast there. There's no indication of any algal blooms. First week of September. I mean, when you get into the middle of September, there are starts of indications of blooms developing, particularly in the south of the Tees area. I think the third week in September, there's a distinguishable bloom, not at very high concentrations, not the levels we talked about, 15 micrograms per litre, probably something up to about 3 or 4 micrograms of chlorophyll, but definite enough to be a bloom of something 2 weeks before the event. After that, it starts to get quite cloudy, and then there's a major storm on October the 6th, which actually brings the material to the shore, and that's the reason that most of the wash-up events, which most of us are quite used to working with. And I should say something about wash-ups in general later on, because maybe that's one of the reasons. Certainly myself, I didn't really think much of this, because wash-ups are quite common. So after that event, a lot of sediment in the water, and we've heard mentioned sediment and chlorophyll in the water compete optically, so it gets more difficult to actually pick up the situation into October. There aren't so many satellite images, but I'm quite confident that throughout September we've got enough. I do weekly averaging of the available satellite scenes, and you can see the development of a small bloom.
So the evidence indicates there was a bloom, and it could have crashed and the dead algae could have sunk to the bottom.
That's normally what happens. So where I disagree is that it was a large Carenia bloom. I don't think it was. I've provided years ago some of the field data that feeds into Peter Miller at PML. He's the one who developed the Carenia algorithm. Been out on the North Sea in a Carenia bloom. They're quite spectacular events. They dominate, for example, the western part of the English Channel in summertime, get large Carenia blooms, easily detectable from space. They are common down the northeast coast of England, particularly in August, off the Scottish coast, probably more from Aberdeen down to the border. They're quite noticeable events. There isn't one off the Tees in September. I'm quite sure about that. The chlorophyll levels are, well, there might be, but it's not a big bloom.
Okay, thank you. Dr. Caldwell?
So which part by answering the same question on the algal bloom?
The question was, do you think the algal bloom could have been associated with the mass shellfish mortality?
So as my colleague was saying, the algal blooms are a natural part of the rhythm of the seas that time of year. It's very common in our temperate seas. You'll have large blooms in the springtime and then a smaller secondary bloom in the autumn. So what we are seeing here in terms of the presence of blooms is entirely expected in the normal pattern of behaviour of life in the sea. At no point has anyone contested the fact that there was evidence suggesting there was a bloom there. Definitely not. The evidence is clear. There was certainly evidence of a bloom there. But that is pretty much all that can be said about that nature of the bloom. You know, that there is evidence that the bloom was there. As CFAS said earlier, there are no cell counts, and there's an awful lot of inference. And I would be very cautious about making definitive conclusions based upon inference. So the system, the North Sea system, is categorically a stress system. There are many stressors acting upon it, of which algal blooms, variable oxygen levels, changes in temperature will be many of the backgrounds, compounded by additional stresses brought about by human activities, whether it's discharge of sewage, whether it is potential pollution caused through dredging. All these systems have a certain level of tolerance to them. Same with anything in life, if you exceed the tolerance of a system, it will break. I think what we are seeing here is an event occurred in the autumn of 2021 that caused that system tolerance to break. And not just break, but break spectacularly. And we have seen through our own eyes and all the evidence that it was the large crustaceans, the decapods, the crabs and the lobsters that were disproportionately affected by this event. Now, that in and of itself is more than just a curio. That warrants investigation. Now, if at this point, Meg, do you mind if I then bridge on to where I think a gap was missed in terms of the investigation?
Well, we're going to move on to pyridine after. Is that so? If that's what you're going to talk about,
linking on to that.
Yeah, we'll move on to that. And Mr.
But continue on your question.
Yeah.
So there was, previous panel put various points that there were toxic algae toxins present in the materials in the crabs, and there are. But here I'm just reading CFAS's own report, and their conclusion is, and I quote, overall there was no clear indications of marine neurotoxins being present in the samples received at the levels which would cause concern. That's
the evidence we got
report.
talked about the sort of asphyxiation of the crab and lobster because of the breakdown of the algae rather than them being toxic algae. I mean, am I correct in saying that's the evidence they gave us? Yeah.
Not necessarily. So the thing is, if it was predominantly down to the oxygenation, it wouldn't just be the crabs we'd be seeing. We would be seeing, in effect, a more or less complete sterilisation of the benthic fauna in that system. You have to ask yourself, why is it predominantly the crabs and lobsters washing up? You would expect to see effectively, it was a mass deoxygenation event, that entire system would be killed. So looking objectively at the picture, it just does not stack up. Well, as I said, this is a complex situation. The North Sea is a very stressed system. There are many things putting stress on organisms, and all it takes is one event. Now, hopefully, this committee will help us understand what the nature of that one major event is.
I think Dr. Foster was nodding his head when I suggested it could be an asphyxiation effect, were you? Or?
I think it needs looking at seriously, and we're short of evidence on that matter. I was quite interested that the CFAS modelling is down that route. That would be one way. So we don't have any in situ evidence of oxygen levels, either surface or benthos, in that part of the world, and we need those kind of measurements. 2021 and 2022, exceptionally warm, settled summers. 2022 broke up earlier, the first storm events in September this year, but last year was quite late. It's quite conceivable, and from our measurements, we've certainly shown in the northeast coastal system there is a stratification of the water, so difference between a warm surface layer and a cold pool below, which effectively is cut off from the surface. We need to find out more about that. There was a study in 2018 from the marine station in Blythe with a glider and underwater robot set offshore from there. That studies the oxygen in some detail going off to the Dogger Bank and back along transect. Some work I've been involved with further out shows oxygen levels can get quite low, below the thermocline. But at this state in our part of the world, it's just speculation, but it could quite easily be something interesting. In terms of the avoidance, for example, herring would avoid areas with oxygen lower than about 25% concentration, simply swim away because crabs and lobsters, less mobile, they wouldn't be able to escape, and because they're larger animals, higher oxygen demand, they might be in the first line of attack and more likely to turn up in a wash-up event. So what didn't get hit by this event is quite interesting. The anecdotal evidence about birds is also interesting, less so from the oxygen point of view, but potentially linked to harmful algae. I was out of Staithes about mid-September last year, and there were dead guillemots and razorbills everywhere, belly up, before bird flu reached the area. Not quite sure if ever there was a proper investigation into that. Most people thought they'd come down the coast from further north, but I've never seen so many dead birds, and that would be round about the origin of this event, about that time.
Okay, understood. Turning to Mr. Dear-Jones, and we'll move on to your pyridine data later, but your report states that there is a lack of empirical evidence to support the government's algal bloom theory. However, the government did find traces of biotoxins in crab tissues that are associated with algal blooms. Does this, along with the satellite imagery and water sampling, not suggest that an algal bloom potentially had an impact? Or have you ruled that out completely?
Yes, I think my position on that was that when I was producing the initial reports, which I did up until June, which have subsequently been superseded by the brilliant work that colleagues on the desk in front of you have carried out, The algal bloom hypothesis came to me after I had pointed out the pyridine issue. And it seemed like, okay, we'll have a look at this. And what it was based on, as far as I could see initially, was the satellite imagery. And I queried the satellite imagery because Plymouth Marine Laboratory, I was sent photographs on a page which had been dispatched to CFAS by Plymouth Marine Laboratory. They had the images and they had a few statements. And amongst the statements put there by Plymouth Marine Laboratory was the fact that the satellite imagery could not define any specific algal species. So it could have been any algae, benign or toxic, as far as Plymouth Marine Laboratory were concerned at that stage. I did additional research into satellite imagery and discovered that also, particularly in the US and Australia, it has been remarked that satellite imagery may mistake algal bloom, the chlorophyll type images. It could be mistaken for surface suspended sedimentary material because a lot of the common analytical techniques applied to satellite imagery needed tweaking and enhancing in order to be able to confirm whether or not sediment was present in that imagery. And I put that through to in a report to the Commercial Fishermen's Associations, who I believe made DEFRA aware of that first report where that was mentioned. And I had asked or suggested that those enhanced analytical techniques be applied to the satellite imagery in the context that the satellite imagery actually was contemporary or nearly contemporary with dredging activity. And as I am fully aware, both dredge dumping and dredging activity itself are likely to create plumes of fine sedimentary material which can rise to the surface, remain suspended for extended periods. And the data I have indicates that such plumes may extend, particularly in terms of the fine sediment material, for at least 20 kilometers downstream. So I questioned that hypothesis and never got the further responses that I was hoping to get, i.e. that the additional enhanced research into the imagery was produced and carried out.
Thank you very much. I'll move on to Ian Byrne now, who's going to specifically talk about pyridine, which we've already had mentioned.
Thanks, Chair.
Pyridine.
Direct this to Dr. Caldwell. Why have your reports identified pyridine as a more likely cause of the mass crustacean deaths than an algae bloom?
Let me put where we came into the study into context first, because I think it's important that the committee understands why I got involved in the study. So whenever I looked at the joint report conclusions from the agencies, it didn't make sense why pyridine was dismissed offhand and wasn't explored any further. Now, if I give you an analogy, it's like if your car breaks down and you take your car to your trusted mechanic, who you've got faith in. The mechanic sees there's a warning light for low tire pressure, concludes you've got a puncture, and then chooses to ignore the steam and oil pouring out of your engine. Where you have such a significant outlier in terms of a chemical showing up in testing, as a minimum, you have to go in with an open mind and say, right, I'm simply going to look at this to rule it out. Can we rule this out of this investigation? If we can, wonderful, we can move on. That wasn't done here. And that is the reason why I got involved, because, one, from a purely pragmatic scientific perspective, that would be the first thing you should do. If you see something, any kind of anomaly, any kind of outlier, you must question it. Wasn't questioned. Second, it doesn't take any longer than a 5-minute search of the scientific literature, even if you're not aware of the nature of pyridine, to think, well, this is not something that can be dismissed without looking into it, because it's a well-known toxin. So that's the rationale of why I got involved in this study. Now, the simple question that my laboratory asked is, can we find out whether pyridine is toxic or not to crabs? That was our very simple starting position. Our hope was we would find it wasn't toxic, and that's great. We could conclude that, and then other sources of stress to the system can be found. However, when we undertook our rigorous scientific work that was done to international accredited standards, we found that pyridine was not just toxic, it was exceptionally toxic to crabs in 2 different ways, which I will come on to. So, seeing the very powerful nature of the activity of pyridine on crabs, it then triggered us to then follow a process that we call a STOP, or a source to outcome pathway, which is an innovative form of risk assessment that we are quite familiar with in our ongoing work in Norway. And we decided to apply that approach to this example. Now, in these STOP pathways, you need to try and find out where the likely source of your contaminant in your environment is coming from. You then need to understand, is that source, is it resident, is it transitionary? What mechanism will turn that toxin, make it available to the organism? What mechanism will allow the organism to interact with that toxin? Does it bioaccumulate? Is it another mechanism? And then how that toxicity actually expresses itself in terms of the crab. And then from there, you can extrapolate to population level effects. Now, before we get on to the source, you need to understand the nature of the toxicity, because it's really quite striking. So we use standard approaches in ecotoxicology where we expose crabs that we collected outside the impacted area. We expose them to pyridine of very carefully defined concentrations along a concentration range. We do this to generate what's called a dose-response curve. That is a standard protocol in ecotoxicology that allows us to then generate lethality, toxicity data from that.
Is that like the LD50?
Exactly, exactly. So we followed the standard protocols, and we assayed all the way down from 100 milligrams per litre all the way down to controls and respective steps in between, and that allows us to generate LD50, LD30, LD10 values from that. And we monitored that over the course of 24, 48 and 72 hours. Now, we were actually taken really quite aback when we did these experiments. We were very much caught by surprise. We were not expecting pyridine to be as toxic as quickly as we saw, to the extent that we were seeing crabs dying within a matter of hours. And you need to understand the nature of how these crabs express the toxicity, and then how that in turn relates to the reports of dead and moribund crabs and lobsters in the field. So whenever they're exposed to pyridine, first of all, depending on concentration, at the higher end of the concentration, they go into very rapid, aggressive, violent convulsions, to the extent that the best way to describe it is the crabs were doing somersaults in the tank. That lasted for anything up to 5 or 6 minutes, whereupon the majority of crabs would come to rest on their back, and then they would enter into a phase of paralysis that lasted anything from 20 to 30 minutes, and that paralysis was progressive. After that period, once paralysis set in, we could still see the crabs were still alive, we could still monitor the movement of their mouth appendages, but they were all dying within a space of 6 hours. Now, that nature of that response reduced, as you would expect, as the concentration went down, but we were still seeing paralysis and death even at concentrations of 20 milligrams per litre. Indeed, at really low concentrations, 2 milligrams per litre, which you wouldn't think you'd see any effect. The crabs were surviving the 3 days, but actually when you handle them, it is as if they were anaesthetized. I mean, if you would never put your hand into a tank with a crab without really being very careful about what you're doing. When you have the crabs that were exposed to 2 milligrams per litre, it is as if they were drugged. They were placid, they were pliable, you could pick them up, handle them without any threat to your safety. And that is the smallest concentration we tested. Now, we looked beyond simply the behavioural and survival data, because that's not enough for us. We want to know what was happening inside the tissues. Now, we did that by sampling the gill, and hepatopancreas and the muscle. Now, I expected, if I was going to see something, I would expect to see signals in the gills and the hepatopancreas. And we were monitoring for signals of cellular stress, a group of chemicals called reactive oxygen species. Now, they're a very natural part of every cell, but under stress, cells produce an awful lot of them, and they cause a great deal of damage. They will attack DNA, proteins, they will damage cell membranes. They're not something you want around in any cell. And that's why we take in antioxidants. The job of antioxidants is to mop up those reactive oxygen species before they do harm. Now, I thought we would see a very strong signal in terms of our ROS, because we're very sensitive measures of this in the gills and hepatopancreas versus the control. I was deeply surprised that we didn't see any change in ROS levels in those two organs. However, in the muscle tissue, we saw striking, orders of magnitude change. So we measured two types of ROS. We measured one called the cellular ROS, which is all the reactive oxygen species put in the cell, and that was a 5-fold increase. And then we specifically measured the ROS produced in the mitochondria, which is the power station of the cell. And that increased by 20-fold. That's almost off the charts. Those crabs then, when you watch them in the tank, they also then tended to drop off their legs and their claws. That all coincides with this incredibly powerful stress signal in the muscles that's causing them to drop their limbs. So you're looking through that range of toxicity from the paralysis, you look at it from the convulsions, you look at it from the mortality, you look at it from the cellular stress, and you start to put together a picture of what might be happening here. Now, it's at this point that I need to get a little bit more academic with you, and I'll try and keep it simple to understand. Because faced with these kind of data, given the low concentrations of pyridine and the fact that the chemistry of the pyridine says it shouldn't bioaccumulate, so with that I agree with what CFAS were saying, it absolutely shouldn't bioaccumulate, why are we seeing the effects that we were? Now, it took me a while for the penny to drop. But then the penny did drop, because it reminded me of something that I teach my own students when I teach them about marine toxins and marine biotoxins. There's a family of toxins produced by a group of marine worms called the bootlace worms or nemerteans. Now, to you and me, they're no great thing. You've got to be a marine biologist and spend a lot of time on the shore to see these things, but they're there. And they are both, strangely, prey and predator of crabs. So they have to both protect themselves and have weapons in their arsenal to take down a crab. And they do that through chemical warfare. They do that through producing compounds. Now, the interesting thing here, some of the most potent compounds they produce are pyridine analogues, pyridine derivatives. And they will pump those into their mucus. So if a crab comes along, grabs the worm, just simply by touching the mucus, that crab knows it's dealing with toxins, and it will abandon the worm. That worm can then go around, attack the crab, inject venom, and then, once it's done that, wonder what the symptoms are. You've got paralysis, you've got convulsions, you've got death. In fact, whenever these first venoms were discovered, you go back to the early French literature, it was initially described as the crab convulsant factor. So all these things are starting to make sense in terms of what we're seeing, in terms of what we see in the lab, in terms of what's been reported by fishers and by public members whenever they've seen moribund crabs wash up. The behaviour and presentation of the toxicity all matches. That still doesn't then address the question posed by CFAS. Well, it doesn't bioaccumulate. If it doesn't bioaccumulate, how can you see those features taking place? And then here, I actually have to say here that I'm grateful to one of the CFAS colleagues working alongside me, one of the toxicologists, to helping us bottom out the actual mechanism for this. The reason why such low concentrations of pyridine and pyridine derivatives can enact such powerful mechanisms on crabs is because the crabs on their own legs, on the outside of their body, have got receptors that have been finely tuned over millions of years to detect pyridine and pyridine analogues. So they don't need to bioaccumulate. There simply has to be pyridine in the water or pyridine in the sediment, and that crab cannot do anything but avoid it. To give you an analogy, we all understand that rattlesnakes are poisonous and are venomous, and we avoid those by keeping well clear. That's a nice, safe mechanism. What were to happen if rattlesnake venom blew to us in a fog we couldn't escape from? That's the kind of scenario you're facing here with the crabs. You've got an animal that has evolved over millions of years to have receptors for this type of molecule on the outside of its body. When it detects those chemicals, it initiates response in its central nervous system, and then depending on the concentration of the chemical there, you can get a whole range of responses right the way through to what we've seen: convulsions, paralysis, and death. So in terms of explaining the toxicity of pyridine, the case is there and clear to be seen. The next step is then understanding how pyridine, if it's released during dredging, could then be transported 50 kilometres down the coast and can enact that toxicity. And for that, I turned to help from my physical oceanographer colleague, who is an extremely talented marine physicist, and he used the most powerful models and simulations available for the North Sea, more so than CFAS has used. And he also then run his model for a month, unlike CFAS's 3 days. And what's more, we have always taken a conservative with a small c approach to our science. We've always underestimated every chemical we use. We've always underestimated all the parameters we use. So we would never run the risk of making false positives. And the same with the modelling. In the modelling, we built into it the actual destruction rate of pyridine. So as been mentioned in the previous panel, pyridine is both lost to volatilization, and it's also aggressively attacked by oxygen. Now, the US EPA have got data for this in the 1980s, and they show that the half-life of pyridine, so losing 50% in water, is 8 days. As I said, we've taken a conservative approach. In our models, we put that half-life to 90. So we would actually remove pyridine from our simulation faster than it actually would in reality. We modelled based upon the release of pyridine on the same scales the CFAS used, so a total release of 10,000 litres. And we ran the model using the tides and the currents affecting the coast on each given day that the dredging campaign took place and for the following weeks. The model clearly showed that pyridine in water would be transported all the way down to Whitby and Robin Hood's Bay. And from that, we could pull out toxicity values, and based upon what we've then generated in my laboratory, we could then make reasonably sound estimates of the extent of population loss from the crabs and lobsters in that given area. So let me start by looking at the epicenter, around about Hartlepool, Redcar, Teesmouth. Based upon our work, we predict that half of the crab population would have died in the first 24 hours. You move further down the coast to Runswick Bay. We predict that 30% would have died within 24 hours. And then, right the way down to Whitby, we believe it would have lost 10% as a minimum of the population there. So we have shown that pyridine is extremely toxic. We have identified the mechanism that means that bioaccumulation is out of the equation. We've identified the way that pyridine will be transported. The next step is then trying to go back to that source to outcome pathway model and trying to come back, well, where is the source of pyridine? I'm satisfied as a professional scientist that I can explain the toxicity of pyridine. I'm completely satisfied that I can defend that. But I also need to identify likely sources. Now, you've heard again in the previous panel that they don't believe there are sources in the Tees estuary. Unfortunately, I've got news for them. There are at least 3 major point sources, 2 of which are historical, 1 of which is much more contemporary. You go right back to, obviously, it was mentioned, the cooking industry. Redcar is internationally known for that. Pyridine as a byproduct is, again, well known throughout the world. It is very well studied, and there's a very high risk level with that. So I find it very peculiar that pyridine is not monitored routinely in the Tees estuary, given what's known right around the world. You also have a source from when ICI were refining crude oil. So whenever they refine crude oil, there is a fraction called the C5 or naphtha fraction that has no clear market value. So they would pipe that to another plant on the north of the Tees, where that naphtha fraction would undergo a thermal cracking process. So in effect, they heat it up to a very high temperature, and they make those chemicals in that fraction form other chemicals. They will then extract from that those chemicals that have a market value. Anything that's not worth them selling was discharged directly into the river. Of course, pyridine was one of those chemicals that had no value to them and was discharged. At a more contemporary level, we also have the company called Vertullus, which had its own specific pyridine and pyridine derivative manufacturing plants. They were using that for a range of products, rubberizers, drugs, herbicides, pesticides, a range of products. There are significant questions to be answered with regards to how they handled their effluent discharge. They were meant to have had a functional wastewater treatment plant on site, and that appears to have never have worked properly. So they then subsequently had to tanker hundreds of thousands of gallons of their wastewater effluent, including pyridine releases, to the Brand Sands wastewater treatment plant run by Northumbrian Water.
Why did we get dead crabs then, when all this stuff was going straight in the river?
Coming to that. So we've got three sources, two historical, one much more contemporary. And now we must then look to the chemistry of the pyridine. As has been absolutely correctly talked about today, pyridine is unstable. In water, it will diffuse widely, it will evaporate very quickly into atmosphere, it will be attacked by oxygen. Therefore, any attempt to measure pyridine in the water is a fool's errand unless you are literally on top of any kind of release. You're always on to a loser in that kind of scenario. But what pyridine does do, when it gets into the sediment, if it's protected from oxygen, it becomes stable, inert if you like. And in that sediment, it will build up over time. And as long as it stays undisturbed in that sediment, in an oxygen-free environment, it's fine, no problem, can't do any real harm. But if you have an event that comes along that allows that pyridine to be mobilized from the sediment, brought back into the system, then you have a problem on your hands. Now, we have measured pyridine, as was mentioned in the previous panel, in the surface sediments down to a depth of 20 centimetres. Now, it's important for the panel to understand that that 20 centimetres would be regarded as the pyridine destruction zone, so that part of the sediment where it will be being lost to the atmosphere, where it will be diluted, where it will be being attacked by oxygen. Simply by the fact that we are measuring pyridine in the majority of our samples in the Tees estuary and the spoil zone is a significant red flag that must be taken further, because it implies that in those deeper sediments where there is no oxygen present, you have a reservoir of pyridine there. And what we believe we were measuring in our surface sediments was effectively the diffusion or the percolation from that pyridine reserve coming to the surface, being interacted in that destruction zone, and that's what we were measuring. Again, simply the fact that we are measuring pyridine in the sediments, given what we know about the chemistry, is a great concern. And that makes it equally puzzling to me why pyridine was not part of the standard risk assessment for any work on being undertaken in the Tees estuary. Certainly any work that involves any risk to disturbance of the deep sediment. And this is where we go on to the dredging. I don't know whether you picked up from the earlier testimony, but it was pointed out on 2 occasions that the extra dredging was in response to a slippage in the Tees. I think that's quite a significant piece of evidence that needs to be explored. How big was that slippage? How deep was it? Was it interacting, interfacing with that anaerobic zone? Is that the mechanism whereby pyridine will have been mobilized from that safe reservoir and then being picked up in the maintenance dredging and taken offshore inadvertently? Because if that was the case, then that's just unfortunate that that's what happened. But that is a tangible mechanism from source to outcome pathway. You had identified 3 point sources of pyridine You've identified the mechanism whereby pyridine would remain stable and resident in the sediment for decades. We've now identified a mechanism whereby an accident, a natural incident, could have caused that pyridine, that sediment, to be moved and to be picked up and transported offshore through the maintenance dredging. We know that this charge of pyridine will have been reaching the whole way down that coast. We've even seen that from the sediment. You speak to the fishers when they put their pots out at Whitby, and they're finding at the time their pots are being covered in fine sediment. That never happened before. So you're not just getting water being transported, carrying pyridine, you're also getting the sediment itself. So the mechanism is there. The toxicity mechanism is there. The mechanism for crabs to respond to very, very low concentrations is there. And then the explanation for them why you're getting this mass mortality within the impacted plume zone and not outside of it. I think you need to understand that. It's a very clear demarcation of where the crabs and lobsters were dying. And when you try and trace that timeline of the movement of our simulated plume down the coast, it coincides with when the washups were happening at each given location. Now, again, someone mentioned earlier that you're still getting crustaceans washing up 2 months after the fact. That can also be addressed. There are, in fact, 2 toxicity mechanisms for pyridine and pyridine analogues to crabs. There's the one I've told you about in terms of the venom mimic, which is incredibly sharp, incredibly acute, and that would have likely have been causing that first major wave of wash-up. However, crabs and lobsters that will have survived that initial exposure, and we know some of them did because we've seen it in our experiments too, they then open themselves to a secondary toxic mechanism because these pyridine compounds interfere with a protein called SARM1. Now, SARM1 has got a really important job in terms of maintaining nervous cells, your central nervous system, and it's functional all the way from bacteria right the way through to us. If that SARM1 protein is switched on or damaged or made to function in any kind of way, it switches from its normal function, which is protecting the nerve cells, to switching around and becoming an enemy, and it starts attacking and breaking them down. So it's a form of neurodegeneration. You know, think Alzheimer's and ALS for crabs, but on a much shorter time scale. Here we're talking about not years for these things to set in. Here we're talking about scales of days. So the acute venom response, you're talking scales of hours to days. For the SARM1 protein, you're talking days to weeks. That 2 different mechanism of toxicity explains the really powerful and very disturbing high numbers of crab and lobster being washed up, and it also accounts for the later wash-ups. We shall have surviving crabs out there succumbing to the secondary toxicity as a result of the exposure. For us to completely and with confidence say I've got a really rock-solid explanation that pyridine is the straw that broke the camel's back in this scenario, we just need to know how much pyridine is in the deep sediment. And we can't get hold of that data. We know boreholes have been drilled. We haven't seen this data. We need to see more boreholes drilling. We need to have an entire pyridine map across the entire depth of sediment in the Tees estuary. There's no point in coming up with any regulatory mechanism based upon monitoring pyridine in the water.
Who's done the boreholes, Gary?
I suspect PD Ports, perhaps. I don't know. I don't know who's done them. We don't have the data. But what I'm saying is any kind of regulatory monitoring based upon water for pyridine is not going to work, because it's just not rounded long enough for it. For you to understand the nature of the risk of pyridine and to manage any given system or any given development accordingly, you need to know what is in the sediment to a great deal of detail. Now, that is the case as we have collected and set it out. Now, what we are not saying is that the dredging and the development around the Tees Port should be stopped. Far from it. We are not the enemy of development and redevelopment of that site. Far from it. Indeed, my own job title is Applied Marine Biology. My job titles are all about extracting value and resource from the sea. What we would like to see by way of a solution is, first of all, a temporary moratorium on the capital dredge until we understand what's happening. You know, the initial pyridine release is done and dusted. It's done its terrible damage. It's moved on for the system. We need to understand whether there's pyridine and other chemicals in the remainder of the sediment that has been earmarked to be dredged out for the South Bank.
Are you just dismissing the algae as the cause of this? Are you absolutely...
As I said before, there are multiple stressors in the North Sea. And I said there would be a strong likelihood, a strong indicator that there will be one major episodic event that will have taken that stress system and broken it beyond repair. My argument is that in this case, algal bloom or no algal bloom, if it's there, and it is there for sure, it's part of the background stress. But what we're seeing with the pyridine story, a fast-acting toxic mechanism that does this terrible, terrible damage, and given by its chemistry, it just runs away. I was mentioned earlier, kind of a thief that hides in the night. It's got that kind of pattern to its chemistry. So we would put forward a range of what we think are pragmatic and sensible solutions based upon the science. We would like to see a temporary moratorium on the South Bank dredge until we do see this pyridine map in place and it has been independently verified, and it must be independently verified. That's really important. From that, we can then develop a sensible and precautionary risk assessment to allow the development to start again. We do not want to stop new jobs, new innovation, the Teesside becoming the next green industrial revolution hotspot. We're all for that. But we want to see that happening without effectively allowing our marine system to be ripped asunder. I don't see there being any real conflict there. We can make sure we do both, but it does take to have that measurement, that full detailed three-dimensional map of the pyridine the way through the estuary out into the spoil grounds. And not just pyridine, but all the other chemicals that are on the MMO's list, and also anything else that's there that isn't routinely monitored. Because who knows if there's another surprise waiting for us. If pyridine wasn't monitored, and we know pyridine is this toxic, we need to take a much more broad, holistic view and ask ourselves, the obvious question, is the suite of chemicals that we test for, is it enough? Should we be looking wider? And on that, we must look to the Tees industrial heritage and understand the chemicals that were produced and released from there. And they should be on that list if they're not already to, and they should be become a really important chemical.
Just on, because I think he's answered nearly every question, so I think we'll move on. That's been a brilliant piece of evidence. So with the rollout of the free ports, is the concerns that this could happen in other areas that are similarly affected from an industrial perspective?
It is.
And I'm thinking Liverpool.
So I'm going to get a little bit of a bleak perspective here. So the autumn event from last year is done. Whatever happened is done. The damage is done. We can't do anything about it. But we can prevent any further damage from the ongoing dredging of the South Bank. And as has been pointed out, we need to look more widely. We need to look at any other dredging activities for any other major ports around the country, whether it be free ports or any other aspect. And we need to really look carefully at the regulatory policies that these things are being granted under. Because certainly from my perspective, they're not fit for purpose for the Tees. It does make me wonder whether they'll be fit for purpose for other estuaries and sites that have their own industrial heritage.
That's a really great comment for the report. Okay, thanks, Chair.
Thanks very much. I think most of your points have been covered in that long diatribe, Geraint, but I don't know if there was any other bits you want to pick up. The only thing that occurred to me is that we got evidence in the first session that we've seen healthy crab with levels of pyridine similar to levels found in the dead crab. And is that something you would expect, or?
That's something that needs to be addressed. So again, I'll go back to the CFAS's own defence in that their method isn't validated for tissues. And so there is scope for them making false positives. Even allowing them, because this is a common criticism that they've put to me whenever I've been talking about the toxicity. even allowing them a 99% error rate in terms of their concentrations, it still has pyridine in the toxic range. Now, just one more final point to add in, if I may. So we've had 3 meetings as an academic group with the different agencies, and over those 3 meetings they have become increasingly collaborative and increasingly productive. And indeed, as an output from the last meeting, I've received a letter from NIFCA representing colleagues from the Environment Agency and from CFAS, and they have asked me to share my data with them, unpublished data, so that we can work together to stop the dredging of the South Bank. And that was used twice in that letter, to stop the dredging. From my perspective, from building up an evidence base, presenting an evidence case, I think what we have done, what we've shown, stands up to scrutiny. And again, applying a precautionary approach, I think my colleagues that have written to me now think that it's pragmatic and sensible that we explore these and we stop the dredging and we undertake further work.
Come to Garrett in a sec. When you say stop the dredging, you mean stop all maintenance dredging?
No, stop the capital dredging. Maintenance dredging, fine, unless there is another slip event. And that's perhaps something that PD ports need to start monitoring. If there are these slippage events do happen, perhaps they need to be approached with a very precautionary principle.
Aaron.
Yeah, so that was an excellent piece of evidence. Can I just ask you a couple of questions? One is, therefore, are you saying that what we need to do is firstly get the boreholes, get the samples, find what is where in terms of under the sediment, at what depths, at what concentrations, and then secondly, until we know that, suspend non-maintenance dredging, and then after that, what advice have you got? Because I mentioned in the previous session some issues about whether there was methods for mitigating potential risk in terms of the nature of dredges and the containment. Is there any advice you've got on that?
So that comes into whenever that risk assessment is taking place. We believe it will be sensible and pragmatic with that information for dredging to be happen safely with the right kind of dredging technology, the right kind of mitigation, including metal sheet piling, that is very effective at preventing release of water and any kind of sediment into the system, whereby you can prevent any release and contain it and take it away from the system so it doesn't get released into the river and as sure as heck should not be dumped at sea. In that situation whereby those mitigation steps and controls are put in place, there should not be any risk. That risk is managed sensibly, pragmatically, but for that to become a reality, we have to know where the pyridine is and where the hotspots are. We don't have that information yet.
And again, this must be the case, as my colleague said, that we should do these boreholes wherever there's a free port, or indeed, in Swansea Bay, we've got this lagoon proposition where we're going to, take out sediment. And your advice would be, firstly, to do obviously the proper testing, and secondly, if dredging is required, then it should be done in this controlled, contained way in a time frame with suction and with walls.
Absolutely. If we don't take a precautionary approach, then we sacrifice one industry for another. We sacrifice our ecosystems, and it's avoidable.
That's probably not possible in an estuary like the Tees with a lot of shipping traffic to put those kind of measures in, I would imagine, where you're doing maintenance dredging of the main channel would mean closing the estuary.
Right. Yeah.
So that would have to be risk-based on areas where there is significant industrial pollution there.
It goes from your mapping, or you potentially. So that map's key to everything, isn't it?
It is. Absolutely is.
I guess what we're saying is, in the event of a free port, if it has to be a significantly increase in the depth of extraction, that must, obviously we need to find out whether there's pyridine in there, and secondly, if there is, that needs to be managed.
Yeah, estuaries have been getting deeper, dredged deeper and deeper. Our ships have got bigger over years and years. I've done a lot of work on the Westerschelde estuary between the Netherlands and Belgium. So the dredge there was from 12 metres. Now, this is not the depth of sediment. This is the lowest water level that's required for a ship to get in. So the big container ship would probably need about 14 metres. So the Scheldt estuary was dredged to 15 metres all the way to the port of Antwerp. And it was quite a controversial thing because it changes the flows in the channel and potential release of contaminants. So there's maybe another source of evidence there to look at more carefully. It's the same for the Thames. There's been a lot of work on the large dredge out of the Thames, which some fishermen claim has destroyed sole spawning grounds and so on. The marked channel, the approach to the Thames is now much deeper than it was. So it's not Freeport's issue, it's just generally the size of container vessels particularly. But the Tees has had bulk ore carriers and ships of 100,000 tonnes for a long, long time. So it's been dredged very deep for a long time.
Yeah. There's issues on the Tyne as well. I remember when I was shipping minister, you have heavy metals. It's one of the shipyards.
With some of the big passenger cruise liners coming in, having enough space in the turning circle on the Tyne as well,
So is there a case to regulate, possibly, the size of ships allowed in certain ports which have got a history of heavy metals and other toxins?
No, the sediment has to be examined properly because it can be disturbed either by the ships themselves, by the propellers of the ships themselves, or by the dredging operations. So that needs to be known. Thank you very much.
I think probably we've had very comprehensive evidence. I know we haven't used Mr. Dear Jones very much, but in your evidence, I remember, was mainly testing of crab in particular, and published results at the period that you got by various mechanisms. I think, Neil, did you want to come in with a...
Yeah, are you finishing off?
Yes, no, I was just getting ready to finish off, but if you want to come in briefly before we do.
Thank you, and thank you for your evidence so far. Your descriptions of the convulsions, paralysis and death, that CFAS told us that they're going to be embarking on algal biotoxin studies in a sort of similar format. Have you or anyone else done any similar studies with algal biotoxins in terms of that?
No, for me, not for those nature of the toxins, for the length of period of time that you have to expose the crabs. I don't have the infrastructure to do that kind of stuff.
Okay, but you can do it in the pyridine side of things. Yeah. Now, you talked about a delayed mechanism of neurotoxicity. Is that something, are your studies ongoing, that you actually then be able to pick up? Are you seeing signs of neurotoxicity in the crabs that are dying in the experimental setting as well, or is this just something that we're seeing?
No, it's very clear. I mean, even at low levels, I mean, you can even see it very visually. You can see that the crabs are agitated. You can see them tapping their legs, and they actually don't have control over that.
Have you seen, is there any sort of nervous system pathology that goes along with that? Have you found
we've not directly measured it, but it's in the literature. I mean, this is old literature.
Have you found that in your current studies?
Yes.
You have. And to your knowledge, the animals that were washed up and dead in when these episodes happened, were any studies taken place to look at neuropathology, to look at reactive oxygen species in the muscle, and the things that you've talked about? Was any of that done?
Not that I'm aware of.
Right. So do you think that's something that could be looked at if things like this happen?
Yeah, if another event happened, but for those type of things, the time is of the essence. We have to get samples very quickly.
Mm-hmm. And if algal biotoxins are potentially involved, could they produce similar secondary mechanisms in terms of free radical release, that side of things? So it could be. But what you're saying is, don't discount the hypotheses, look at both. And
as I said, there are many stressors in there.
Yeah,
but the very striking nature of the pyridine is seems to be the one that's broken the system.
Thank you. And just finally, Chair, Dr. Foster, you talked about some deaths of birds as well. In your studies, in your sort of group of universities, have you seen any evidence in other animals like seals, porpoises as well? Anything that you can share with the committee at all?
Anecdotal evidence on seal pups, I think.
Yeah, so this is data we're trying to get hold of. It's quite difficult. So there's a population of seals in the Teesmouth called seal sand. And every year there's a report put out by an organisation called INCA, which is funded by Teesport. And the current report said that they had a bumper year in terms of calving, and it seems, on face value, seems they did. I can't remember the exact number, it was in the 40s. However, there's maybe only 4 or 5 of those pups actually survived. So you had a lot being pupped, but the survival rate is incredibly low. And those that survived, were in very, very bad condition. We are at the minute trying to pull that data together independently, but it will take time for us to do that. But we are looking at it.
If we talked about some of the difficulties of measuring pyridine in tissues subsequently as well, is there any grounds for, with marine mammal strandings or bycatch or any marine deaths? I just say this because we have a parallel inquiry on marine mammals at the moment. Yeah. Is there any grounds for sampling in some way in terms of dead birds or dead seals?
And what I would say, from what I've understood from the pyridine chemistry, it's unlikely to have had that effect on vertebrates. It seems to be really very highly evolved to target crustaceans, but I'm not ruling it out. But I would say it'd be less likely that pyridine would have been the cause for that. It might have been a background stressor, but I'd probably say there'll be other causes for those. Okay,
and I know I'm jumping around the species a lot, but I declare an interest as a vet. Have you had any sort of discussions? I know the University of Liverpool, the Small Animal Surveillance Department, had looked at, there were some worries about dogs that potentially were having clinical signs of vomiting and diarrhea, and I think they concluded that perhaps was not related, and perhaps it was a circulating viral gastroenteritis or something like that. Is that? Do you have any dialogue with other academics?
No. We haven't expanded the conversation any further beyond that.
Okay, thank you.
Before we finish, Robin,
it was just thank you, Chair. Just to pick up on the point that you're making about maintenance dredging and capital dredging. Do you, in terms of sampling for capital dredging, are you aware of any sampling that is happening before capital dredging was taking place, or in your view?
I don't have those data.
No. So if that, so therefore, leading on from that, do you think that data is important to have before coming to the conclusion that capital dredging should be something that doesn't take place, and that maintenance dredging should be allowed to continue?
Yeah, I mean that's the point I was making.
Yeah.
We need more data. We need to understand what chemicals are present in the sediment that will be dredged and discharged. Without that information, it's quite a silly thing to do, to risk releasing chemicals without understanding the nature of the risk.
And therefore, if we had that data, then we
have that
have
data,
more knowledge on building mitigation.
Mitigations can then be put in place, but you must have the data. You must have that level of understanding first, and we don't have that yet.
Okay, I think the evidence we've got is that sampling is taking place, which is why some of this material will have to be disposed of on land, but they're not testing for some of the chemicals that we're talking about today.
Yeah, but I would like to see kind of that data be made more publicly available and actually taken independently. I think sadly a lot of the different agencies now, they have a bit of a credibility issue, and it's desperately sad for me to say that, considering many of my students now work for different agencies. And I think that credibility gap needs to be worked on and repaired going forward.
Okay, thank you very much indeed. I think it's been very helpful to hear basically two scientific theories which we're trying to test. And I think the key, I think we'd all agree we need more data. We need to try and see what is happening, and if future dredging takes place, it needs to be done safely. Thank you very much indeed. Appreciate your time and all the work you've obviously put into this subject as well. And thank you, Mr. Dear Jones, as well. I know we haven't used you a lot, but I know I read your paper last autumn actually, and was when it was drawn to my attention. Thank you very much indeed. We now move on to the third panel, which is, I think, aimed at the discovering just how profound the effect has been on some of the fishing communities, and given that some of those in my constituency, I know exactly what we're going to hear, and it's not good. Thank you. So if we can ask our third panel to come up.
Amazing. That chat. Absolutely amazing.
I'm Joe. Can you sort them out, please?
Yeah. These have brought some free fish.
So, welcome to our third panel. I must say that two of the witnesses, Mr. McCandless and Mr. Redfern, are old friends of mine. In fact, Joe, we were together on last Friday, weren't we, in Whitby? So, but for the benefit of the rest of us, could you introduce yourselves and say what your particular role is? Starting with you, Dave.
Yeah, thanks, Chair. I'm David McCandless. I'm the Chief Officer of North Eastern Inshore Fisheries and Conservation Authority. Now, I'm not sure if the panel are aware of the work of IFCIS, but but we are the inshore fisheries managers of the English coast. Our particular area covers from northeast Lincolnshire up to South Tyneside, and a little bit about my background, which I think is relevant. I'm an academic. I have a degree in marine biology and oceanography and a master's in fisheries biology and management. I've worked in the region for 26 years, and 22 of those as chief officer, so I think I'm well qualified to hopefully answer questions to the panel. Thank you.
You go out to 12 miles, and most of these problems have happened within 3 miles of the shore, is that right?
Yeah, currently 6.
6?
6 miles.
Thank you.
Yeah, I'm Joe Redfern. I'm the Secretary of the Whitby Commercial Fishing Association and General Manager of the Whitby Lobster Hatchery. I've got a degree in marine biology and coastal ecology and a master's in marine biology. And I've been working in the fishing sector. Indeed, I worked for the IFCA and David McCandless a few years ago. I've been working in the fishing sector on the North East for the past 5 years.
Thank you. Mr. Rennie?
Good afternoon, Mr. Chair. Thanks for allowing me to speak today to the committee. My name's Stan Rennie. I've been commercially fishing for 48 years, from part-time with 2 jobs to full-time the last few years, and I'm here to represent the fishermen, coastal communities and the North East Fishing Collective guys and ladies on the marine dives.
I think before I start on the sort of questions that we've prepared, maybe it would be appropriate to ask what your comments would be on what we've heard so far during the first 2 evidence sessions, maybe starting with Joe, picking on you.
Yeah, I mean, it's been a long year, really, since this happened, and we've gone through many different iterations, I think, of what we've thought as a fishing community, and, very thankful to Tim Dear-Jones, really, who pointed us firstly towards the dredging theory way back in January. And it's very amazing, really, to see the evidence that particularly Gary's provided today, where there seems to be such a growing body of evidence that I think it's now undoubtable that the dredging and pyridine has played a part in the mass mortality events that we've seen. And I think there's been a lot of inadequate science and done by the agencies, unfortunately, over the past year. And yeah, I could go on, but I'm sure Stan will have some more to add.
David, and maybe you could sort of say what stocks are like now, because you'll be monitoring what's going on.
Yeah, it's as Joe said, it's been very challenging since October, and I think where our organisation is placed as the IFCA is kind of right at the coalface. So if anything does happen in the coast, we're normally the first point of contact. And we started getting those reports in October last year of shellfish being washed up in the inner Tees, and then reports of gradual spread of issues down the coast. And those issues stretched down to as far south as Robin Hood's Bay. They didn't quite touch Scarborough. And to the north, there were reports up to Seaham Harbour as well. So very concerning, and as per usual with these sorts of events, the first consideration is a pollution incident, and that's kind of how the whole, I guess the whole response started from that, that the epicentre was very clearly the Tees, and there was some pollutant factor, and in that respect, then the Environment Agency, as the appropriate organisation, stepped in first to commence the investigations, and then as the whole event widened, more agencies got involved with the expertise and knowledge we don't have. We manage fisheries, and that is where our expertise is in assessing stocks and managing inshore fisheries appropriately. So our role was very much to support the government agencies, collecting water samples or anything that they requested, gathering intelligence. there's a central intelligence unit which is managed by the MMO, and all intelligence was fed into that, and all the reports that were drawn in. So, yeah, very challenging, very demanding. Where the stocks sit, we have done a piece of interim work, and I have seen it waved about in social media that kind of, with some people suggesting that it says things are okay in certain areas, which clearly, if they think that, they haven't read the report or read it clearly enough. And
we've seen some landings in Hartlepool which are from further out.
Yes, indeed. There's been a major spike in crab landings, particularly at Hartlepool over the last couple of years, to the tune of about 800 tonnes, and that shows up in the national statistics. But that fishery is directed out in the central North Sea. It's larger nomadic vessels which have other ports of operation. They're not based locally in Hartlepool, and they fish out in the central North Sea, out in the Dogger Bank area. There's a very good series on the BBC at the minute which is worth a watch, because it does give you, a really good insight to the challenges that the fishing industry faces, and some of the fishing vessels involved in that fishery featured in the first series of that. So that has caused challenges in terms of us, just to take a wee step back, in terms of us trying to assess where the health of the fishery is placed. Primarily, we've used government-derived catch and effort statistics. So licensed commercial fishing vessels must submit catch and effort data that's gathered by the MMO. We've used that primarily, and that right there is the challenge, to try and decipher out those ambiguities and try and get to the bottom of where the stock is. Our most reliable catch and effort statistics come from Redcar, because Redcar is primarily inshore boats between 4 and 8 metres in length. They're almost exclusively inshore, so the catch and effort data from Redcar is the most reliable at the minute. I have to say the report only covers up to July. We've supplemented it with doing some of our own survey work, so we've worked pots from our inshore fisheries vessel. I've put staff out with some of the commercial vessels that are working in the affected area. They're gathering data from the catch, and some of the staff have been out with Mr. Rennie, and then quayside observations and some questionnaire-based information. So overall, the broad regional picture for lobster is reasonably encouraging at this very early stage. At broader regional level, it seems to be following seasonal patterns. Obviously, as we progress towards the summer, catches increase, they drop off, as you would expect following that. They're within sort of average tolerances of the data we have available. However, there is a big question mark over crab. Most notably and most obviously, the impact has been in the crab stock, and I think that's borne out through the evidence presented across the day today. We think we know most prevalently to the north of Whitby is where the impact is most significant. Looking at the figures, we think landings are at least 50% down on where they should be in that area. Perhaps in some cases they're maybe higher, maybe up to 90% down. And it is having a big impact. Now it's impacting more the smaller vessel, the smaller section of the vessel fleet, so particularly the 4 to 7 or the 4 to 8 metre vessels that are far more restricted. They've less flexibility in terms of where they can fish. They can't move very far offshore. So they're much more restricted to near shore and inshore. I'm sure other evidence in the panel is much more tuned into that. But that is our broad assessment at the minute.
Perhaps I could turn to Mr.
Yes.
Rennie and give us some indication of the impact it's had on individual boats, individual fishing families, and how people are faring, actually, because we are very concerned about these traditional fishing families and their businesses.
Mr. Chair, prior to that, could I get the opportunity to answer the question what you thought of the presentation?
Well, we listen to the evidence and we will then discuss what our response is, and I think we've already come to a few conclusions, but we will be, we take evidence and then we consider what we need to do. We hold ministers and government to account, and that's what we'll be doing in this situation, have no doubt.
I'll talk on how it's affected local catchers. I find it so frustrating that NIFCA, and me and David were in a public meeting last week, and unfortunately I'm going to say the same thing. NIFCA's report is so misleading, so out of touch with reality what's happened on the die-offs in the die-off zone. Why NIFCA, who's in control of 6 Mile Out, would include nomadic super crabbers working up to 4,000 pots each, fishing 90 mile off the coast onto the lands of Hartlepool to try and say the landings have gone up for the local fleet. It just puzzles me. We've lost half the Hartlepool inshore potting fleet in the last 12 months since the Orca dredge. So half of the fleet have been forced to the wall. Boats sold, or there's still a couple for sale. Jobs lost, land jobs looked for. I've got a very good friend, Paul Rutherford, who I've known all my life, fished alongside him for the last 30 years. He has 2 boats and he had one where he was doing up. When the Orca dredge occurred, I can imagine what difficult situation he felt to sell in, where he had to lay 2 of his sons off and a crewman what had just trained up because there's just no money to be made. I'll give an example of the catches, totally different to what NIFCA says and how people find them. But Paul's catches, his lobsters are 50% down, undersized lobsters 75% down, buried ends, pregnant lobsters 75% down, velvet crabs 100% down, short crabs 100% down, brown crabs 100% down on the catches of the previous year.
There's nothing there, in effect, crab-wise, brown crab, nothing there.
No, not where he's fishing.
No, and it's centred round the Tees. It seems to be the epicentre. That's what we've seen. So as you come further down the coast, you start to see some of the brown crab, but they are 80-90% down. But none of the velvet and the shore crabs are completely decimated on the area. But the centre and the epicentre seems to be focused around the Tees estuary and around Hartlepool.
I mean, a few years back I went out with Gordon Quinn, who you may know, and we found that for every marketable lobster we found, there was probably 10 or 12 undersized. How long does it take a lobster, if those have been taken out as well, how long will it be before the lobster that you're putting in the sea from your hatchery become big enough to be landed?
5 to 7 years of growth from a juvenile lobster to a commercially landable lobster, which has to be 87 millimetres across its carapace. It's 5 to 7 years. So any sort of measures we put in place now, we're going to see a lag phase until we see the impact.
Understood. Rosie, I think you wanted to come in.
Thanks, Chair. Yeah, you've covered a lot of this, but I think I particularly wanted to put this to Stan. I mean, you might not think that in Whitstable and Kent we have a huge amount in common, but my fishing industry has been decimated by things like sewage and the outfall of the pipes, and I totally feel for you that you've been doing this 48 years and you've probably never seen anything this devastating, I would imagine, to your way of life. So sounds like a simple question, but if you could spell out to everyone watching how the local communities have adapted and are people leaving the industry, because presumably no one's going to want to come into an industry that feels this fragile.
It's so heavily regulated now, fishing. We've got no young recruits coming through. It's so difficult. This year, I've only got a small boat, 6.92 metres, 26 foot. I normally fish in front of the window and on the doorstep. The wife keeps an eye on us. Fished Lunsker Rock for 47 years and me 48 years with my own boats. My catches are down 80% lobsters. I rely on the summer season, July to September, to get the majority of the money in, what will get me through the year. I've been putting, well, my earnings last year in that period, my turnover was £8,761.84. This year it's £3,753.21. A 58% reduction, but that's not just a 58% reduction of what's left. My working days more than doubled. I'm trying to stay. I would never land anything what I wouldn't eat myself, and neither that Teesmouth, I certainly wouldn't eat anything till I made sure it was safe. So I've stuck to my principles. I've tried to get out the die-off zone this year. I've been steaming an hour and a half a day in a little boat 7.5 mile north towards Seaham, 3.5 mile off, putting weather away what I shouldn't be in, like the deadliest catch some days out there. And it's just devastating, absolutely devastating. And it's not just, it's the full ecosystem, it's the full marine ecosystem. There's nothing in the pots. There's none of the squat lobsters and everything we enjoy to see. We've tried to fish Lunsker sustainably for donkey's years, a lot longer than before buried ends couldn't be landed. I actually got 2 warnings off NIFCA for V-notching my lobsters and letting them go, because at the time you couldn't land a V-notched lobster, and myself and another
people
friend...
maybe there's a programme that the government ran. If you catch a buried hen,
yes,
you put it back in, you notch its tail, you get paid for that, but if anyone else catches it before it catches it, it sheds its shell, you're not allowed to catch it.
No, you don't get paid for it. You put a V in and it can't be landed.
Yeah.
So the fisheries were buying lobsters off the buyers to V-notch and return. So the likes of me and my colleague has put literally tens of thousands of lobsters in the last 20 years, what we could have landed, V-notched them, buried ends, put them back, trying to build the stocks up, be sustainable. And there's more other people doing it. And there's more lobsters about around that area up till 25th, 26th of September last year than there was when I was a kid. First starting off on my own boat in 1974, 13-year-old. It's had a devastating effect on the coastal communities. As I say, we've lost half the fleet. There's boats down Whitby, that's all they've known fishing. We've got a proud heritage. We're proud people in the North East. We fight to the last. It's not right where we've had to rely on donations from the public and the likes of the Fishmongers Company to get this independent testing done. But how can we rely on the agencies who've come up with fabricated...
just before you go on, just with the V-notching, there's boats out of Whitby that have been V-notched 1,000 lobsters and returned them, and now they're coming to me and saying, has that sent them to death? You know, has the pyridine made their way into the blood? Because they haven't seen any of those V-notched lobsters again. You know, so there's this real feeling of hopelessness within the fishing community, because you know they're trying to do what they can to protect it, but they're still just not seeing anything coming back. So you know the well-being and the mental health impact has been really dramatic along the coast. A lot of people just felt there's been lack of support, lack of trust, hopelessness in the industry, and sort of betrayed by the agencies as well.
In the cost of living crisis as well.
Exactly, yeah, and after the pandemic as well, where we were severely hit with that. And so much so that the fishermen's missions now started doing like regular health checks in the ports that are affected, and offering charitable hardship funds, which has all been raised basically money through our own campaigning.
So you feel really, really let down, and that there's nobody supporting the community. Presumably, is your gut feeling with your experience between you that the community has changed forever, or do you think
my heritage fishing goes back on my mum's side? 400-500 years they've traced our family back, fishing in front of my house on the beach. I think I'm going to be the last one. And how can we trust agencies? We've got independent investigations being undertaken and they're coming out with a totally different thing. I didn't find the first lot of evidence very good, but when we get the CFAS and the Environment Agency reports coming out, on the 6th of September last year, I reported a pollution incident when I was going down a boat. It happens regular. There was a scrap ship load in Hartlepool. It was a flat calm day. The the scrap dust was coming all over the headland. It was going in the water. When it was getting near the quaysides, it was sinking, so the water was ginger about 2 foot deep. So I reported at the pollution hotline, like I've done dozens of times, and they know what it is. So on the CFAS and Environment Agency report, they include one of my photos from the 6th of September of scrap dust pollution in the water, to what I can only think is trying to push their theory of red algae.
It says there the suspected red algae bloom that they've used is...
So I've got all them on my phone, and all the evidence I sent them on a day. I didn't get any feedback.
I think just
who can we trust? We need independent investigations.
Come back to your point as well. I think there's just a lack of diversity in the region now. You know, we used to catch whitefish and herring, and you know, a more variety of fish. And Dave McCandless can talk further on that, I'm sure. But today it's just only crab and lobster. You know, that's basically 99% of the boat, and it's the mainstay of all the income for the North East. So, you know, it just shows how sort of fragile and vulnerable it is, and something event like this, the dramatic and drastic impact that it's had.
Yeah, I need to correct you there, sorry, Joe. It isn't just the crab and lobster fleet, it's the prawn fleet. Prawns are shellfish. When the Orca dredge had started last year and finished after 10 days, the prawn fleet were catching dead prawns round the dumps and the clay all the way. If you just bear me, I've got an email from a boat, 2 boats actually, from the last couple of days. They went to sea for a 10-hour tow, and they got a grand total of 4 prawns each. So it's not just that type of shellfish, it's all shellfish and lots of marine creatures.
That's going to cost them, isn't it?
Cost them £300 to go to sea for the day to make nothing.
It seems to be that crustaceans, decapod crustaceans.
Yeah, so I've just worked 7 days a week when I can get to sea, because of you further out, 7 days a week, 8-9 hours a day, to cost me over the summer quite a bit of money to go and do it, to try and get some money coming in.
Thank you both very much.
Thank you. Robbie?
Yeah, thank you, Chair. So just following on, I just wanted to touch on government support. So it's our understanding that UK government haven't provided support to help fishermen and women affected by this problem. Is that the case? And has there been any local support that's been provided?
We've had plenty of local support of the likes of Jill. We had meetings with Jill. Jill and one of the lasses who works with Jill, Sally Clark, volunteered to do evidence document what's been put in. So I was collecting and sending all the evidence in, and that's quite comprehensive. Some of the local councillors have been brilliant from day one, supporting us from day one, because we knew there was something not right. My catches fell off a cliff. The Orca dredge started on the 25th of October. 27th of October, we knew there was something wrong. I was thrown back 40, 50, 60, 70 buried hens a day, and the ones what I hadn't caught before, V-notching them. So that's a day. I haven't caught 70 buried hens this year to throw back. I had it together all the summer. So I was doing that a day.
If you're talking about financial support, we've had no financial support from government or from local councils whatsoever. The only sort of financial support that's been in place, as I said, has either come from local businesses such as Whitby Seafoods and been given to the Fishermen's Mission, which is a charity which has then allocated hardship funds since that.
So the government have mentioned making use of the Seafood Fund. So I was just going to ask what your thoughts were on that.
Well, it's not the fund in place for hardship. It's not for fishermen. It's for innovation. There's 65 million out of that 100 million is for infrastructure. You know, it's for scientists. It's for local authorities to try and improve the innovation. There's no allocation from that 100 million whatsoever for hardship, and certainly not in response to the event that's happened in the past year.
And most of that money was allocated last year, wasn't it?
Yeah. Okay.
So it's not new money.
So what other support would you want to be put in place by from the government?
I think there should be more grants in place for some diversification, you know, to try and help people to start looking at other fisheries, you know, and to try and you know, maybe just move away from the reliance on crabs and lobsters in entirety, you know, to give the fishing industry more options really on that front. And that's for those that are able to do that. It's probably more for the boats, further out of the impacted area. For places, I'm sure Stan can go into this, but around the Hartlepool, a decommissioning scheme where those that simply can't make a living, are given the compensation to cover the debts that they might have on the boat, to cover the debts that they've got on the business, and to provide that support while they sort of transition out of the industry, because they haven't got a choice.
That's the only financial thing. We've had 2 proposals from that. I think one was in the meeting that Jill was in at the Fish Quay. And this is how desperate it is. People were suggesting decommissioning plus where they'd get decommissioning plus some losses of what they've experienced and not being able to go and say again. That would give them 2 opportunities. That would either let them get out of the industry and clear the debt and the boats would be chopped up and the government would keep the licence, or for the smaller boats, it might let them buy a bigger boat where they can get out of the die-off zone safely and fish. But that's just going to put more pressure on the ground, but that gives them an option to make a living, and that's all we've been suggested. We've never once went to a meeting asking for handouts. If we've been offered food or anything, if the lads had been offered food, they wouldn't have took it. They're proud people, so not once has it been mentioned in any of the meetings we've been to. Yeah. Only thing what's been mentioned is buy the boat off people, chop it off. And
is there anything else that on that point, David, that you wanted to expand on as well?
Just to add a bit more to what Joe and Stan have said, there's a lot of fundamental work at national level that needs done to restructure the fishing licensing system, access to other species in terms of quotas, etc. And it's going to take quite a few years to sort that out, but Joe's absolutely right, and it's one of the fundamental issues of having been in the common fisheries policy and the challenges of being in that in terms of restrictions around quotas to target whitefish species, etc. And as a result of that, over the years, in our region in the northeast, most of the inshore fishermen are operating on capped licences, which means they only have a very limited, if any, take of species such as cod or haddock or whiting. And pretty much, as Joe said, those vessels are stuck now in 365 days potting for lobster and crab. So when you get an event like this, which is unforeseen, and certainly something I've never experienced in my career, which is long enough in that region, you get something like that no one foresees, and the fishing industry is under immediate and serious pressure because they can't diversify. They can't, for example, bring some pots in, set nets for seasonally for cod. Even when I started, which was in 1996, we still had some inshore vessels that worked lines for cod in the winter, and all that has rapidly disappeared, and the fishing industry are just so reliant on potting for shellfish, and to say that it's under huge pressure now with something like this that no one could have foreseen, and they do need a leg up. There's no doubt about it. There's no doubt about that at all. The one thing I would say, having sat through listening to the other 2 panels, I've been in all of that work myself. We've supported the investigatory work through all the government agencies. I've been in, at the minute there's weekly meetings with the government leads, etc. I've been all through that. I've sat through 2 or 3 ministerial briefs as well with different changes coming through. And my personal opinion is there is a genuine will to try and get to the bottom of what has happened. I've got to take my hat off to Joe and the fishing group. It's amazing how they've managed to pull together the collaborative research, and Joe has done that from scratch. He's actually done that from scratch, and that's no mean feat to line up 3 universities or 4 universities with different experts, fund that, self-funded to do that research, and there's a lot of good work going on there. What I've found where the IFCA sits, and it's where I think we are pushing hard. We kind of sit between all the cracks here, between the different government agencies, and we've been working very hard to try and keep the collaboration going between the fishing industry, research that is being led on behalf of the fishing industry, and the government research. And I have to say, it isn't easy, and I think if we weren't pushing to do that, so we've established kind of a joint technical research group that draws in all the leads on both sides. We've had 2 meetings of that group, and we'll keep going with that. And it's like banging heads together sometimes, but there is a genuine will right across to try and get to the bottom of this, and we need to get to the bottom of this, because this would decimate the North East fishing industry completely if it happened again. And there are so many gaps in knowledge at the minute. We really need a precautionary approach going forward here. Very much so. Thank you.
Thank you, David. Yeah, and I mean, while we're trying to collaborate and to get to these answers, the fishing community is completely terrified that if this next round of dredging goes ahead, that there's going to be nothing left, and that's going to be the complete nail in the coffin. We're absolutely terrified, and that's what we're fighting for, is to prevent that round of dredging until we're completely sure that there's no toxins, there's no impact. And hopefully you've seen today that we are not completely sure. With the algal bloom theory, we're talking about maybe we haven't seen anything like this in the past 50 years. But from the satellite imagery and from the data, it doesn't seem to be a once in a 50-year algal bloom that we've seen. We've seen algal blooms like this over the past 50 years, but we've never seen this sort of impact. So, we are really, I can't stress it enough, terrified that the next round of dredging goes ahead, that it's going to be the end of the North East fishing industry.
Carrying on from one point David said, and it's shown fair, the important thing for the small fishers, the under 10 boats, is diversification. So for all the time, most of people's lives, you wouldn't target stock what was down. So if there was plenty of crabs and lobsters on the ground in the winter, you'd carry on fishing. You'd leave the cod alone and the cod would come back. Then just out of the blue, it's happened, I think, 3 times. The MMOs picked a reference year. So if you were diversification, say from potting to netting or longlining, and you'd spent the full year at it because there was plenty of quota there, the catch was there, and you left the potting alone, so that was growing them stocks up. Then all of a sudden, 3 times it's happened, they pulled a reference year out the window, backdated it, and if you hadn't landed so many crabs that year, or lobsters, you got your licence capped, so you couldn't go fishing for crabs. So then that was them boats stuck on the fish, and then when the fish disappeared, and there was plenty of crabs, and it was the same for the fish boat, couldn't diversify on the crabs. So what we've ended up is lots of people getting the licence capped and not being able to diversify. And getting on to a point Joe just mentioned about the Freeport dredging, I had a film crew up the river last week, and we are terrified. And something, the ecosystem is under an extinction threat for it must be a couple of 100 square miles in our area. There's been a migratory lobster catch in the summer, which you would expect, coming into cast, coming into mate, then going back out. So people's had a reasonable summer, but I wouldn't fish there because you don't, neither Teesmouth, you don't know where it's safe. They're going back now and you're stuck to the local stock. So whatever's left of them local stocks, there's nothing going to replace them next year. Your pots are normally teeming with undersized lobsters and everything. So next year there's going to be even less, and the year after, and it's going to be 7 years before we see any improvement. But the other thing is, our spawn from the lobsters goes down the coast quite a considerable distance. So in 7 years' time, they're not going to have a generation to fish to. It's got such a huge knock-on effect. So I was up the river with a camera crew a week ago. I'm sorry for overloading you with emails, but I sent a load of evidence in, but one was a link to some footage what Joe edited for me. The Freeport dredge started on, I think it was the 1st of September, and it's all supposed to be contained and going to landfill. So if you looked at that footage, what you would see was a barge with a huge excavator on, with the teeth of its bucket, and a flat plate what comes over the top. So he's lifting the dredge from the bottom, this toxic dredge, mind, from the bottom, and it's spilling out and it's dropping in the river, and he's putting it in a barge next to him. And when that barge is full, they tow the barge quarter mile up the river, and there's 2 excavators emptying it, and it's dropping all over the place. There's no silk curtains, there's no nothing. They're opening them seams up in the most toxic place where they said they'd found the toxins. Seams up to the tides in the spates of water, and we're terrified it's going to be a tip of the edge occurrence again.
We have already started to see some like more wash-ups.
We've had wash-ups since the 1st of September, haven't we?
And sort of like in the weeks that have gone on since then seems to have,
and
that's material that has to be disposed of to land because it's proven to be toxic.
But we had a meeting with the mayor and we asked him if he would test all of the testing samples for pyridine, and he wouldn't, and he refused when we asked again. But I cannot believe that the licence application and the subsequent licence being granted does not contain any reference to the Orca dredge or the die-offs. The risk assessment doesn't, the environment impact statement and the method statement. So the live documents, we said this, the live documents, if you found something on the day, you would add it to it and you would counter it. And there's no mitigation feature in place if this happens again. So, it wants to be cofferdammed off. It should be cofferdammed off the river. That river must be wide enough to get a ship past. Jerry could answer that. To get a ship past to go half the way across and dredge it out and take it away without anything escaping, and then do the same for the other side. But we're terrified our industry could be lost now. It's heartbreaking. And it's not just the industry, and it's not a monetary thing. It's our ecosystem. We try to look after it. We enjoy seeing everything in there. We try and share it with people. I've never kept a big lobster in my life. I've never kept lobster over £6, but I do like to bring them in and show Sarah, the daughter, or anyone's about, and wait and put my book and V-notch it and let it go again. And all of them lobsters we've put back and all that money we could have earned is laying dead on the ocean floor in the North East.
Yes, did you a quick? We are getting very tight on time. So very quick.
I just thinking. I know Stan said you weren't looking for compensation, but I mean, it does strike me that if you had just by way of example, an investor in a country where the government came along and stopped them working, they could sue that government for the expected profits or revenues in the future years. And it does strike me what you're saying is that had this event not occurred, that you could have looked forward to a sort of lifetime of income, and there is a case to be made, given the evidence we've heard, that this was an avoidable disaster.
Yeah, it is. But what I am saying is, if somebody says to me, Stan, I'm sorry about what's gone on, here's £150,000 or your ecosystem back, we know what we'd pick, our ecosystem back.
Right. I mean, on that point, then obviously a lobster takes 5 to 7 years to mature, as you've just said. I mean, what would you like to see happening?
I think first off, the pollution, must be stopped. Any sort of measures that we trying to regenerate, the ecosystems, we've got to make sure that we're not putting any more pollution or damage in it. That's just simple. Whatever we do, we've got to make sure that it's safe to do so. But there are sort of measures such as like habitat regeneration. And sort of artificial reefs, and like bioengineering, like there's a lot of industry, such as the wind farms going in around the area that are going to be rock armouring their cables. They could all be made in a way which promotes, reefs, promotes life. And Redcar Council, and from my obviously personal perspective, sort of things like stock enhancement with hatcheries, can go to start putting something back, basically. But I think it's very important that we've got to make a stop to the pollution first.
I agree on hatcheries, but it's just the tip of the iceberg, but everything else. But the different creatures what's been killed off, and Gary would tell us if I was right, but it's as though they can take the pyridine for longer than other creatures. So we ended up with the crabs and lobsters disappearing overnight. But we've had wash-ups of everything you can think of, from sea urchins, razor clams, worms, lots of seals dying and porpoises washed up on the beach this year, but everything you can think of in the marine environment is washed up. And we don't know whether you'll ever get it back because there's other creatures taking over now. The pots are full of little tiny whelks, absolutely covered from 7.5 mile north to the inshore waters around where I work, where you can get out and plodge half the time. And never seen a brittle star in my life, so I've never worked Lonscough for the first time in 48 years this year. I went to sea with Paul Woodfield a couple of weeks ago. His crew was busy, so I went to see to work his pots. He's been doing a survey in Lonscough in the die-off zone. And I couldn't believe it. The pots were absolutely covered in brittle stars. So the marine environment is changing already. My baits, even though my bait was in bait bags 7.5 mile away from the headland, which is 3.5 mile away from the Tees mouth, trying to get out of the die-off zone, the lampreys, the devourers, absolutely covering the pots, and it's as though there's an explosion of that bottom of the food chain, and it's going to change things for the worse, you know. So will we get our marine environment back?
Finally, can I ask David, because David, you talked about collaboration between Newcastle and Hull, etc., and DEFRA, but it seems to me what we've heard is essentially a DEFRA cover-up or a failure to investigate Pyridine. Isn't there a risk that if there is a sort of collaboration, that there'll be an attempt again by DEFRA to cover up the emerging evidence of Newcastle?
Like, I personally haven't seen any evidence, any direct evidence of anybody trying to hide or cover up, and I say that genuinely just from, as I say, I've been heavily involved throughout from day one and across all the meetings and discussions that that we've had. I've always seen that as the way to get to the root of this, and the only way is for everybody to collaborate, the industry-based research and the government research, and forge that stronger collaborative approach to get to the bottom and learn more. And there's so many gaps in knowledge.
I think so. It comes sort of back to a point that Mrs. Duffield made in the first panel, I think, where she asked about trust, and I think unfortunately Jerry answered that because he's not a non-governmental agency. But there's sort of been a few events, I suppose, over the past year, which has kind of led to that. I think this is maybe what you're talking about, or maybe where our perspective, as we're suffering from this, really. I think it was the first public meeting we had was on the 18th of November with Dave McCandless and his Deputy Chief Ian Davis, and the main takeaway we got from that meeting is they told us we should go and collect our own evidence. We can't trust the evidence that we're going to be presented by them and by the agencies. Go and collect your own evidence. And that was a powerful thing to say, really, and surprising, but that's what led us on to Tim Dear-Jones and basically pursuing that and getting our own evidence. At the end of January, we presented that evidence to DEFRA, and we said, we're going to give you a time limit because, we're kind of a little bit through this, and we want to know what your response is going to be to Tim Dear-Jones's dredging and Pyridine theory, bearing in mind that we didn't know a lot about it then. There was none of the work from Gary. It was just a theory. So we presented that to DEFRA and gave them till the 4th of February to reply. On the evening of the 3rd of February, there was a big press release came out from DEFRA. Investigation was closed. Algal bloom theory is the most likely cause. And that's the only correspondence we got to. I had to email them and say, is this press release a reply to our correspondence we made?
Unbelievable.
So I've never ever accused anybody of a cover-up, and I don't want to, but from our perspective, where we're struggling like that, and you come up against these things where this Open Democracy article had found emails that had said they were trying to push through the algal bloom theory as a rebuttal against the dredging theory. It's it's a struggle, really. One more point I just wanted to make as well, and it's just really about the Pyridine theory and just the similarities that we've seen in the industry. We've seen exactly the same symptoms with crab and lobsters, and not only just the immediate symptoms of the convulsions and paralysis, but also the long-term impact. So in June 2022, so this summer, the merchant in Whitby reported a 2,000% increase in dead lobsters in his tanks, and the same with the Andy Beavers. And everybody's been reporting that there are weak and they're sort of showing these long-term symptoms. It's exactly the same geographical area as shown on the dredge plume model from Gary, and it's exactly the same pattern of mortality. So it's in the plume model that Newcastle University produced. The plume starts outside the Tees, and it takes 2 weeks to make its way down to Whitby. That is exactly what we saw. Every single symptom, every single event that we saw happen in the Tees made its way down to the coast, and we experienced it in Whitby 2 or 3 weeks later. So there's just so many similarities between what's been proven experimentally by Gary and what we observed along the coast.
I've got to admit, yes, I have accused people a bit of covering up. When I get reports from CFAS and Environment Agency with a photo of an incident I reported much before the die-offs, when we get reports that crab stocks are up and stuff like that, when we know they're not, there's just an email here from a merchant. The mortalities, he would estimate approximately 10 to 15 kilogram a month we expected, less than 1%. But the animals that were weak when brought in fishermen would be put in a hospital tank where more often than not they would be revived and put back into the stock. Towards the end of May, we started hearing more and more reports of weak dead lobsters. When the boats came into land, they had to physically inspect every lobster. They were having to check them every day in the tanks, and they'd lost 200 kilograms, this is one buyer, in one tank, that they'd had to pay the fishermen. And there's no recompense for them. You know, without the merchants, we've got no living anyway. And we just feel like we've been ignored. You know, if it was anywhere else in the country, we feel like the agencies would have thrown the kitchen sink at it. And with the back of beyond up the northeast, you know, no disrespect, but half the people of the country think England finishes in Leeds, but it doesn't, goes on to Berwick. And we can't be forgotten. This North Sea is an extinction episode. It just hasn't had anything done. It hasn't had the commitment of the agencies to come up with theories. No, it's not good enough.
It's the biggest lobster fishery in the whole of Europe along the northeast. And for each of those jobs at sea, there's 7 jobs that they support on land. So it's a huge fishery.
How many jobs are you talking about, Joe?
What would you guess, David? And I think Whitby, you're talking about 150 connected. So further up the coast, that's just in Whitby alone. David probably has a better estimate. I'm not sure.
Yeah, being that sort of region.
There's a lot of the fleet being lost in Hartlepool. What with the licence capping, the EU boats coming in and plundering the seas and not getting our waters back what we thought of. We just hope that it will recover. But
and I did read this morning there was £1.1 billion was compensated to the farmers for foot and mouth disease in 2001. 1.1 billion to slaughter those animals. So, you know, it's
I'm conscious of time. We've actually, I want to apologise for going over time, because I think it's important we hear every single word of evidence that we need to. Neil.
Thank you, Chair. And I think my final question, I think you've answered a lot of it, so I will wrap it up with a sort of lessons learned sort of final question. But thank you for your powerful testimony. And a lot of today has been talking about the worry about the ecosystem. But I was very struck, Joe, I think you first brought it up, in terms of the impact on people. And it really overlaps closely with our mental health inquiry that we've been doing. And we've been looking at acute episodes. You mentioned foot and mouth, infectious disease outbreaks, flooding episodes, that sort of things, but also chronic stresses in terms of livelihoods as well. And it seems to me what we have here, we have an acute episode, but what you have said, it's ongoing in terms of the fish stocks as well, and the stress on people. And you've mentioned the word terrified a number of times in terms of anxiety. What are your local communities doing to support each other through this? And then a recommendation for us to government. What can government do to help people with this real pressure on their mental health?
Yeah, so the first question, what we're doing as communities, I mean, first off, we formed the North East Fishing Collective. That was never a group before, and that really gave us that platform to work together and support each other, really. And we reached out to people like the Fishermen's Mission, to sort of amp up the support from them in the area. But you're right, it's been a massive impact. People just worried sick, losing sleep, unrelenting stress, And there seems to be a knock-on to like physical health as well, you know, to people along the coast, because there's so much at stake. And the second question about what government can do to support, I think is just putting those assurances and support packages in place to make sure, the people that have been most impacted by this have got the support in place and the support from the government.
It's been devastating to people. But you've got to look at health and mental health, haven't you, in the same vision. I might be pleasantly plump, but I'm a grafter. But when the October die-offs came and I had to bring the pots in, and there was nothing in the nets, you'd normally get lobsters and whatever in the nets. There was just nothing to be had. So for about 6 or 7 months, I wasn't working. I was waiting for the spring lobster fishery to show, which it didn't. Then the summer lobster fishery to show. And then because you're in activity where you used to be grafting all the time, suddenly you get high levels of diabetes setting in. And then now I've got back fishing for the summer, even though it's out the district, and I've made nothing. It's back down again. But the mental health, I try to keep from the north end. I might bombard the North East Fishery Group with emails. People send me emails saying, can you keep us up to date with whatever's going on, because it's a big worry for us all. So I'm up till 2, 3 o'clock in the morning sorting emails out. I get up on the morning, my wife's been through everything. She gets up at 5 o'clock. She's been through everything. My daughter Sarah, the mental health of just my wife and daughter, what this has had on them, never mind the fishermen. And for someone in my position, with no training, having grown men phone me upset on the phone, and I'm trying to give them guidance, it's harrowing, absolutely harrowing. And that's how desperate it's been. And people having to sell up, forced to sell up, one of the lads had just bought a load of pots to go potting a couple of weeks before the die-offs occurred, and he had to sell the pots, and his boat's for sale and still for sale, but they're still suffering that anguish. They can't get shot of the boat, some of them. My half-cousin, Philly Cole, he had to sell up. He's a potter, he had to sell up. It's just terrible. And we need to ensure that this does not happen again anywhere else. And we need to ensure that it doesn't happen again with the Freeport dredge. What the Port Authority and the Mr. Ochin and his crowd might be saying, that wasn't a Freeport dredge, but what they can't say is that it wasn't to accommodate Freeport ships, that Orca dredge. You know what I mean? So it just seems to us that that has tipped the ecosystem over. All we're asking the government for is help. Help for the people who's affected, help for the ecosystem to somehow try and recover.
Thank you. Sorry.
Thank you. That's very powerful. Thank
Rosie,
you.
one last.
One really short question.
Short point.
It is. I know this must be incredibly difficult because you're sort of proud working people, but is there a way that we can tweet out anywhere that people could donate to the community if they wanted to? Is there like a, you mentioned a fund.
Yeah.
Is there anything that we could push out
we
there?
do have a Just Giving page, that has sort of been paying for the ongoing investigations that we've been doing, and also, been used as towards like hardships as well. So yeah, there is a Just Giving page for the North East Fishing Collective.
Thank you.
And we've got to thank everybody who's donated to that page. We wouldn't have been able to get these independent investigations, and we wouldn't be where we are now. There's some really generous people and companies. As I say, we mentioned the Fishmongers Company. We've had anonymous donations into that fund of like £2,000 and £1,000. The fishermen, the crews, the families have put anything in. We've said, there's lots of people who can't afford it, they've suffered hardship. And if they had a spare 10 bob, well, even that 10 bob counts, you know what I mean? And that's how we've got to where we are now. And I can't thank all of them people in the public enough.
Yeah, but I do think, that said, and we are very grateful for that. We are going through a cost of living crisis, and we shouldn't be looking for people to give us charity. They should be coming from government support.
Good. Well, thank you very much indeed. I think, the scientists can argue, as we've heard already this afternoon, but what you can't argue about is the profound effect that this disaster, this ecological disaster, has had on fishing communities and those on land supported by those fishing communities. And I know from my own constituency, which is Whitby, Brunswick, those ports which do rely on fishing, particularly for crab and lobsters. So I think as a committee we need to think very hard about what we've heard. And I know, when this was going on, I was talking to the Fisheries Minister, Victoria Prentice, almost every single day. What are you finding out? What do you know? And I think we need to, now we've got a new Secretary of State, I hear Therese Coffey is the new Secretary of State, I think we need to ask some very important questions to them and find out what more can be done. And I think there is probably more research that needs to be done to find out what exactly happened and not just draw a line on it. So, Stan, you can have the last word.
The last thing I would like
I'm
to
ask... I'm Hartlepool, yours are the last word.
Gary, I was shocked last week at the public meeting at Hartlepool, but just to show you how it's affected the whole ecosystem, could you give the results of your square metre survey?
Yeah, so we've been running...
It may not be picked up by the microphone, and it's not usual to have somebody else, but if you want to just come and sit in David's seat for a second.
We need to hear this.
Yeah. We've been maintaining with Newcastle and Liverpool universities a long-term monitoring data set called the Markland data set, and we monitor a number of sites around the UK. This is its 20th year. And we analyse and collect data once a year in the summertime in August. And we haven't had time to go through all the data yet, but we have pulled out the data just for the barnacles alone. Now, barnacles, if any of you know anything about marine organisms, are about as hardy an organism as you can ever hope to get. In fact, there's a billion-dollar industry developed to find ways to kill them. So they're really hard as nails. Yet, there is a virtual extinction of the barnacle population at the Staithe site, which is halfway down the plume. Sites to the north and to the south are unaffected, but there's a virtual extinction and there's a complete loss of recruitment. Now, this event happened and coincided with the natural breeding cycle. So not only have the adults at the Staithe site been killed, the entire next generation has been wiped out. So
it's also due to note crustaceans as well. So
I think we'll have to draw stumps there. And thank you very much indeed for this panel. You've been absolutely clear what the problems are, and we will certainly, as a committee, consider what further steps we should take, having heard that evidence. Alex is going to brief the committee after we... can members remain, because we're going to have a little discussion about what we can do from here. So if the members of the public and the witnesses would like to leave, we're just going to have a little discussion.
Really appreciate it.
Thank you very much.
Thank you very much indeed.
Thank you.
Thank you.
The proceeding has ended. 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 The proceeding has ended.